Good morning, everybody. Again, good morning. My name is Rob Furl. Together with Dan Dombacher, we co-chair the committee for the midterm assessment for this decadal study, the decadal from the National Academies that guides this portfolio of research and helps NASA understand the community's input to the portfolio of science that they oversee. The midterm committee is in the middle of its work for the calendar year, and this particular meeting is one of the major ways in which we solicit and collate community input to the committee for our assessment of the progress of science in this group. The committee is identified by blue badges. If you see people in blue badges, feel free to knock on their heads and tell them what you think. The whole purpose of this meeting is to gather your input, not just to listen to the people up front here give their presentations, but through question and answer and probing and conversation, we hope to gather as much input as we possibly can over the course of the day. Please don't take any questions that the committee puts forth as any sort of conclusions yet. We're at the beginning of data gathering, and so our questions will be on the nature of probing, not on the nature of giving you hints about what any conclusions there might be. We have 6 panels today that constitute our view of 6 areas that we wanted to pay attention to today. There's no way we could cover every bit of science that's been done here. So what we did was we asked for each panel a group of 3 speakers to come on in and give lightning talks limited to 10 minutes that are meant not to cover all the data in the field, but to ignite discussion. And then the moderator for each session will moderate that discussion and invite questions from the floor to keep the discussion going throughout the periods assigned to it. The participation today by all of you folks is important. I want to thank the American Society for Gravitational and Space Biology for the board joining us today. That society is deeply involved in this portfolio of research, and their participation here is both welcome and enjoyed. I hope they are the source of many of the questions that will come to the floor today. To get our discussion started today, and realizing that science, especially in the world today, is all about context, we've invited Dr. Gail Allen to present to us on the state of the exploration architecture, which draws heavily upon this group of science for enabling deep space exploration. Gail's the Deputy Chief Scientist from NASA, and I'll welcome her to the podium now and thank her for joining us today. Is this okay?
Mm-hmm. This advances? Yeah. Okay. So thank you very much for inviting me to talk this morning. I guess my knothole in the agency now in the Chief Scientist Office has given me a little bit of perspective across the agency on science. So what I wanted to do today was give you a little bit of a perspective. Obviously, I don't have the answers, but I have some thoughts that I'd like to convey so that maybe it's a good way to stimulate the conversation for the rest of the day. So as Rob said, one of the things that's really important is how does the science play into the future of space exploration? So one of the things that Bill Gerstenmaier has been conveying to the National Academy, National Advisory Council and so forth, is the approach that we want to take for getting to Mars. Our endpoint is still Mars, humans to Mars. And as you can see in this rendition now, this is very similar to I call it the squid chart that we used a little bit earlier. The same phases, but a little bit more content behind them. So in phase 0, we're still talking about what, if you were familiar with it, Earth reliant. In phase 1 and 2, we're now at cislunar. Phase 1 is to conduct missions in cislunar space. We want to do some tech demos and some science that we think we need to do beyond low Earth orbit. and to develop the deep space gateway and the deep space transportation system that will carry us on into Mars. Also in cislunar, phase 2 will be getting ready and going forward on a shakedown cruise for Mars. And then, of course, phases 3 and 4 will be humans to Mars. One of the things I want to comment on, and I know I don't have much time, so I'm going to this is going to be warp speed, but I wanted to comment that internally we are doing, Sam Shamimi and I are working on an action that is looking at long-term research with demand side, what we need within the agency, whether it's research or technology, and we're asking the questions to the directorates within the agency is what do you need to have done, where do you need to have it done, and when? And so can the lowest common denominator be an Earth analog that you need the research done? Does it have to be low Earth orbit, and if so, does it have to be the International Space Station? And also, if it needs to be beyond low Earth orbit, what absolutely has to be done as far as the lowest common denominator on that? Because at the Deep Space Gateway, which was what we were referring to a few months ago as the habitation module, there will be limited research. We're going to try to put the capability on that platform that needs to be, but it's really a platform for doing very minimal amount of research. So think about that as you go forward. Where do we have to have that research done, and what absolutely has to be done beyond low Earth orbit? One of the highest ones we'll talk about a little bit in a few minutes is radiation, obviously. This just gives you a little bit of a text for what I was just saying, but like I said, space station, it'll end with the testing. We don't put a date on it. We want to make sure we've got the research that we need completed. Cislunar is the testing. We want to do some tech demos, and also do the shakedown cruise I was talking about, and then move forward into sending humans to Mars in the 2030s. Not going to go through every detail, but I just kind of wanted to point out that you'll see the phases. So EM1, we had a study, the study for EM1 and crew, which EM1 was supposed, and that's exploration mission 1, was supposed to be uncrewed the way we've planned it. The administration, the president, asked us to look at whether we could crew that. There is a study that was completed. It's been briefed to the acting administrator, Robert Lightfoot, and to the White House. It's not available publicly yet, and we're waiting for a go-forward plan. The second one here, the SLS, the Space Launch System, could actually carry the Clipper, which is going to Europa. There are pros and cons on that, so we're really looking at the trades, which doesn't really apply directly to us. But here I wanted to mention, this is where we're going to start building that deep space gateway. It's the first one is power and propulsion, the second one is the habitation module, logistics, and then the airlock. And the idea is that this gateway will be available internationally. So we're trying to make it general. We're trying to make it generic enough that when we have international partners, for instance, if ESA or any of our partners want to, other space agencies want to land on the surface of the moon, that may not be our plan, but we want to have that gateway available for them to be able to do that. And also the gateway would be the platform where we would do the research beyond low Earth orbit. And I just won't go through the rest of it, but as you can see here, the point is that the gray here is the Orion, so that would be the gateway, the deep space gateway, and Orion would be able to attach to it as needed when we wanted to crew this. And the idea for the gateway, the habitation module, is that it's a 30-day mission. It isn't manned or crewed 24/7. They would be missions going back and forth for about 30 days at a time, and that's the game plan right now. And it's being, developed and matured as we speak. And that's the other EM1 and out, and how they plan to do the shakedown and the Mars transit. You have the slides. I'm not going to spend a lot of time on that because I really want to leave this open for questions and answers. And this obviously is really what we're about. It's the biological and physical sciences and the research that can be done on the space station in space. And then the big question is, why do we need to do it? What do we need to do, and why do we need to do it? So that's what I want to spend a little bit of the time that I have on, is thinking about this. And earlier on, we had a discussion about push and pull on research and technology. I think that there needs to be a portfolio that handles both of those, that addresses both of those. The pull for this for biological and physical sciences will come, in my opinion, from two activities within the agency. One is the NASA technology roadmaps, and the reason that I want to kind of emphasize those is that they were actually reviewed by the Academy. and we were given feedback from the Academy on areas that we ought to focus on. And so these are the areas for the technology roadmaps. But as you can see, there's space and power, and these are just arbitrary. I mean, there were others: human health and life support, and then over here the materials. They were just ones that I kind of wanted to use as an example of areas where a pole could come from developing in the physical sciences as well as biological sciences in microgravity. So from the roadmap, which is like it was open-ended and everything we wanted to do, we've actually honed that down to the NASA Strategic Technology Investment Plan and have narrowed it down to these 10 technology areas. So looking at physical sciences, and I just want to kind of re-emphasize that for me, I think some of the basic physical sciences we do in microgravity are the TRL below zero, that is the discovery research that feeds into potential capabilities for breakthroughs on technologies that we're going to need as we go forward into space. So that's why I see this as a pipeline, the physical sciences as a pipeline going into technology. And you can see the areas here: life support and habitation, robotics and autonomous systems, lightweight space structures and materials, entry, descent, and power, landing and power. And I just want to kind of spend just a little bit, and this is, I know, an eye chart, but what I tried to do is, so this is what the Academy reviewed, the roadmaps as well as the technology plan, and I just highlighted as examples a couple of them. This is the liquid cryogenics, and these are the ratings that the Academy gave. So a liquid cryogenic is a low and medium risk. But what I did then is I went to the decadal recommendations and started pulling from those recommendations on things that would apply to those areas like that. So this is just not how to do it. It's just an idea of how to think about when we go forward. And we're trying to have these compelling arguments of why we should be doing this research and what it's going to give the nation as we go forward. Another example is the propellant storage and transfer, and I put ZBOT because that's an example of the pull. Not only is ZBOT, which is a cryogenic project that we do within the life and physical sciences arena, but it has been pulled by the space tech as being a capability or a discovery science that they need to be able to help with their cryogenics. So that's what I'm talking about as far as the pull. Not in stone or anything. These are just my ideas and my thoughts, but standing where I am now in the Chief Scientist's office, I'm looking at a couple of things. One of them I will mention is that, from Science Mission Directorate, the science community that we have and how supportive they are and what they do in the modeling of that, maybe compared to what we're doing in life and physical sciences. One other example, which again is this on the left here, is what's in the technology roadmaps, and this on the right are areas that I pulled from the decadal recommendations. But it's long duration health, behavioral health, human factors here, and radiation. And obviously radiation is key throughout this, because not only do we need that on the ground base that we do radiation, but that is one of them that we really, areas that we know we're going to have to focus on beyond low Earth orbit as well. And then what do we do in the habitat? What capability do we put there to be able to do the radiation research that needs to be done? And that's why you have to kind of think about all of these. It will not be, again, I want to emphasize, it will not be a space station capability and at cislunar it will be minimal, but those things are going to be critical. And the compelling story we put behind what needs to be done at cislunar is really key to making this successful. And then, of course, on the right are the decadal recommendations. And I just pulled, these are examples, again, not end all, it's just the examples, but a thought process on the pull side. And you all have seen this. This is the human research risk, path to risk reduction. And I want to applaud, actually, the space biology folks and the human research folks just had a workshop to come together and determine how space biology can support some of these areas that are still knowledge gaps within human research. And so if space biology can focus on the basic knowledge gathering, the scientific discovery, it will definitely lead into and help the human research program as we try to mitigate these risks. And so that is a pull, I would say, from the biological side. So I just want to say, and I have one more slide, but which was like last night, I was laughing, telling Rob last night I was thinking about what we need to do, and I have one more slide. But this, I just want to say that we need push and pull. Scientific discovery can actually be both. The roadmaps in the human research path to risk reduction can be really helpful on the pull side of knowing where we can actually fit in and how we can add scientific knowledge to help move forward in that. Push areas, we need push areas as well. And I know Craig has been working diligently on this push and pull. I think he calls them adapters and some, but okay. But I want to say that there are areas, for me, like right now, if you talk about it, I'm just going to say in an engineering mentality, a critical path is something that has to be in the critical path in order to get the job done. So some of this push technology or push research, plant research, I'm not sure you can justify it's in a critical path. I think downstream it will be, but that's the whole idea about the push, is that it may not be able to communicate or convey now what that value is, but it's going downstream, it will be extremely valuable, and we do not want to let go of that capability, and we don't want to let go of pushing forward in the discovery on the plant research. There are many others like that, but there has to be a rationale and a justification. We obviously can't do it all. I want to just say, right now it looks like our budget for the next 5 years will be flat. That isn't even an inflationary aspect to it. It is flat. So at a meeting I was in yesterday, Robert Lightfoot said that flat budget without inflation being considered, it brings us down over the 5 years $3.4 billion in buying power. So obviously when we go forward, we need to have a good story, a good reason why we want to invest in these research areas. And I'm just going to put one plug in here. We did in 2012, after the decadal came out, we looked at the two founding policies, enabling and enabled by, basic research and what would enable us to do exploration. And we took a very systematic approach. We looked at the weightings on it. We try to be as objective, take the emotion out of it, and try to come up with a go-forward plan. And our go-forward plan actually showed these areas, the light blue right here, there were 12 high-priority recommendations within that decadal that satisfied both the enabling and the enabled by. So we thought that that was a good way with the limited budget to go forward. So again, that's another way to look at this. It's not the answer, end-all answer, but it was one of those things that I just wanted to communicate that it is, there are ways to go about this that we can actually have these compelling rationale that we can convey to the stakeholders, because it's your job to kind of identify what those research areas are and how we should go forward, but it's all of our jobs, including NASA and everyone, to be able to put the story behind it that conveys it to the stakeholders and that is compelling. So my last slide is, last night I was thinking about this, and I went back to the 2003 biological and physical research implementation strategy on how we were going to go forward. And one of the things that we had in there that I still think is it may not be the same questions, but are compelling questions. These were organizing questions on why we're doing what we're doing. And I think that one of the things you need to think about as we go forward, and after we get the results from this study, is how we communicate that, how we package it, because that is the extremely important piece of this. So think about why we're doing this as you go forward today, why we're doing it, why we're spending the money, because again, as you well know, and when in tough times, and I'm talking about the flat budget tough times, technology development and basic research seem to be the banker for everything. So we don't want to be the banker. We want to be the one over there pulling the money from the other side. So help us figure out the compelling story. But I would say that right now we're doing great research, amazing research. The platform is there and we need to use it. We just haven't packaged this well enough to be able to convey in an organized, systematic way why the research we're doing is needed and why there should be a no-brainer when we say we're doing X and why we're doing X. The stakeholders should say, yes, we know that's exactly what's needed. So these were some of the questions. How can we assure the survival of humans traveling from Earth? And then there are some breakout questions. How does the life respond to gravity and space environment? What new opportunities can research bring to expand understanding of the laws of nature and enrich lives on Earth? And again, one of the things I think Craig is going to be, Dr. Kundrad is going to be talking with our chief engineer. Several years ago, we put a line in the budget for foundational engineering science. The chief engineer's office is looking for tools and techniques, but I was from the chief scientist area was pushing for also basic physical sciences, where I can go back to the TRL 0 and where we can do the discovery research that will help in these areas that we need as we move out in space. What do we have to do, and where do we have to do it, and what still needs to be addressed from a research standpoint? Technology we need to create, and then how can we educate and inspire? But anyway, that was like 2003, but I think that these questions are still compelling, and the questions are still there I'm not going to say the general public, all of our stakeholders, whether it's Congress or the President's office or our public stakeholders, all of these can understand when we're trying to answer these questions. So as we go forward, just ask that you think about this as you move forward and we decide what we really need to be doing as we go forward. And that's it. Thanks.
We have time for questions and answers. The microphone is either there in the center, or we have the throwable light coming up from the back. There's a microphone here. Right. The floor is open for questions.
Thank you. Richard Jordan from the Royal Academy of Science International Trust, representing Her Royal Highness Princess Dr. Nisreen Al-Hashemite, who is a scientist. In terms of the last point, number 5, to educate and inspire, we have created at the United Nations, through the delegation of Malta, the International Day for Women and Girls in Science, to inspire and to educate young women who may or may not be going into sciences of all kinds. So February 11, please put it on your calendar, and we are happy to work with you to inspire women in science to do the pushing.
Thank you. Thank you. We will definitely get in touch.
I guess I have the catch mic here. On the chart number 3, so that kind of, for me, is just not NASA. It's all of national goals. So my question is, how is NASA intending to involve some of these other government agencies, the NIHs, the NSFs, in addressing, if we're going to kind of use these questions to kind of guide us? How are you interacting with some of these other agencies, to maybe number 3 in particular?
So I would say that really we're working with the Life and Physical Sciences Division. I mean, that's really a thrust for them. We're there to help with the communication and the bigger picture. But that's the intent, is to have those discussions on a broader note. We're kind of hoping, to be honest, the venue that would be best for that, right, is the Office of Science and Technology Policy. We have the Life Sciences Subcommittee and those things. Right now, until it gets settled, we don't have that infrastructure in place, but that really is the way, because, I mean, for the Life and Physical Sciences, or the Life Sciences Subcommittee, that I was a member, and as Craig was, there are like 25 agencies, federal agencies that are there, so you can get the message out in a very succinct way. But right now we're working through that. But that is the desire, is to get everyone on board.
Hey, good morning, Gail. Erica Wagner, Blue Origin, here with the ASGSR board. Could you go back one slide? So I'm really curious just to hear your thoughts on how we do number 3 here. What is your thinking about how we have a portfolio approach that prioritizes exploration, but still preserves the opportunities for discovery-based science and things that may not be on that critical path today? Can you speak a bit more to that?
So I keep going back to the plant research, because that's the easiest one for me to be able to convey. And obviously, if we're planning to go to Mars with humans in 2030s, I'm not optimistic that we're going to have a capability to grow our own food. But there are other reasons for having plant development and research on the station. If you recall, Scott Kelly has said in numerous interviews that for him it was the tactile aspect of that. It's the indirect value of having capability like that on station. Downstream, it may possibly be that we are actually able to produce food, to grow it and eat it. But for right now, that's the bridge that I see, is that there are other compelling reasons other than in a critical path, and that would be one of the examples. So that's the hard part. I wish I'd had the answers for that. But there are areas like that. It can't be everything, but if you can think, and again, it's that big chasm between knowing what we need and then these capabilities that we have out there, and how do they fit into the big perspective of things. That's the hard part, and I don't have an answer for that. But looking at the gaps that we have in technology, the gaps that we have in the biological and human risk, and being creative in thinking about the capability and the value our community brings with all of their innovative ideas to be able to address that, and just the compelling story about that. I think that's the most important thing, is how we tell that story to convey to our stakeholders that this is something we should be investing in.
Do you see it as a set aside within the portfolio, a certain amount, or programmatically? How do you
I think set-asides are always risky. I think that the story should be integrated, and that we have a balanced portfolio of these pulls and pushes, and any researcher and stakeholders will understand that. ONB understands that we don't all want to have to be going down the path, and shouldn't be going down the path of having a pull all the time, that we should have that push, but they need to have the story to help justify it. And that's what I'm asking you all to be creative, excuse me, in thinking about how we communicate that. And what those should be, because it can't be all of them.
Dr. Allen, in about 3 hours I'll be addressing your item under organizing questions 3, I call it 3B. There was no B, but the question was, how does structure and complexity arise in nature, which is a very broad and cuts across many different areas. This work that I'll tell you about came out of the shuttle era, and the science actually has been moved along over the last 20 years, but not under any kind of NASA encouragement or support, though I've been encouraged by your so-called PSI program, where this kind of very expensive early data are being archived. My question is, to address and get people activated in something like question 3B is all well and good, but with your last comments about the projections on budgets, flat, inflation not included, et cetera, how realistic is it to get people encouraged, enthusiastic, and wanting to work with NASA? Because, as I'll point out, these early studies done back in the mid-90s, 20 years ago, were just cut off at the knees when budgets got a little rough for NASA due to the ISS over expenditures in the early construction phase. So people have been burned very badly, and many of my colleagues simply have told me, I'm not working again with the expectation of reasonable support.
Yeah, it's excellent. I know. I've been in the world of OPPR and the ups and downs over the life of the program. And the point is that we know that we have huge technology challenges and biological human risk challenges going forward. And the point is that we need to discover, we need to do the research to give us the scientific knowledge to be able to bridge those gaps or fill those gaps. For instance, I'll give you the asteroid redirect mission. Okay, so this budget, it was zeroed out, but the point is that the technologies and the demonstrations that were in that mission are not going away. We're still going to do solar electric propulsion. So the mission went, but those are the kinds of things where if you have the compelling story that this could help you identify the gap or fill the gap, those are the things that can be pushed forward. But that is the compelling story, and I can't emphasize that enough.
Thank you very much.
Thank you.
We have to move on to our first panel, so let's thank Gail once again. Well, the first panel is getting set up. I want to remind everybody that this is being webcast, so when you do ask a question, if you could take just a minute to identify yourself and your affiliation so that people know where the question comes from, and be sure to, whenever possible, use the mics. I'm going to turn the floor over to Gail.
Great, another Gail in the room. So I'm Gail Wallace-Jack from Northwestern University. I'm on the committee that is evaluating the decadal study. It's really my pleasure to welcome our three panelists this morning, and I want to thank them for taking time out of their busy schedule to be here. So this is the radiation panel, and we're focusing on questions about mitigation of radiation risk, the role of other risks, not just cancer, but also cardiovascular disease and CNS risk, and dose rate effects, and what we might expect to encounter in a Mars-type mission. I'm going to introduce all three panelists at this time, and then ask them to stick very closely to their time, and then we'll have time for questions afterwards. So the first speaker in the panel will be Catherine Held, Dr. Catherine Held. And I should mention that all three of these are esteemed because they are all either present or former presidents of the Radiation Research Society. My list says that, Frank Cucinato will be the first speaker. Frank is from, but let me, no, you're going to sit, you're supposed to give your talk from here. But I want to introduce all three. So Frank Cucinato will be from the University of Nevada. He worked previously with NASA. He will be our first speaker. Dr. Catherine Held from Harvard and also from National Council on Radiation Protection will be our second speaker, and our third will be Charlie Lamoli, who's from the University of California at Irvine. All three of these are esteemed people in the radiation community. I think we can be pleased to have them here. So Frank, please go first.
All right, thanks, Gail. So we're short on time, so I want to hit some major points. My first point is in the title about relevant doses. A lot of the things you see that make space radiation sound scary is when you look at research results is people are using way too high doses in their studies. So I want to clarify that in my talk. Okay, so the risks that we're worried about are acute radiation syndromes, but we can dismiss that right off the top. And that's because this chart here shows this was reviewed by the National Academy in 2012, a model we put together, the statistical model of solar particle events. This is in gray equivalent, so there's an RBE, a relative factor, compared to gamma rays of about 1.5 for protons. The highest solar particle event you can get with light shielding, 5 grams of aluminum shielding, is about a gray. So on spacecraft, you have 20, 30 grams with no problem. So you can get these doses down below 0.2 gray. So unless there's an operational failure, there is no acute radiation syndrome risk on any space mission. But you do have cancer risk, cataract risk, central nervous system effects. Looks like it could be a problem, but it's not conclusive. Circulatory disease is a problem, but it's much smaller than cancer risk. So we did publish from the astronauts several papers showing that they do get cataracts from low doses of space radiation. So I won't talk about this in my talk, but it brings up the issue, would you get a cataract during a Mars mission? So if you extrapolate these results from lower doses to higher doses, it becomes plausible. So the problem with space radiation really is the types of radiation. So we call it HC particles, high charge and energy. They have both quantitative and qualitative differences with X-rays and gamma rays. So the quantitative differences is handled through the RBE or the quality factor, but the qualitative differences is an unknown. Do they cause effects that X-rays and gamma rays do not cause is the issue. So, and then since there's too few astronauts, except for something like cataracts, to do a study, these mostly were worried about late effects. You have to do ground-based research to get the answers. Okay, so this is a track showing you where the qualitative difference comes from. This is what we call track structure, how the energy is deposited on a microscopic scale, nanometers, the size of proteins or DNA. So this orange part, you can see how high the dose is in that core. So X-rays and gamma rays never have a core. You would have to have about 1,000 gray of X-rays where everyone's dead immediately to have that high of a dose deposited in DNA or protein. So a very, very low dose. It's a single particle like an iron particle in the cosmic rays or a silicon particle has this core, so that's where the qualitative difference comes through. So that's the issue. Now the doses, the other thing I mentioned is the doses people are using are too high. Solar particle event doses could be higher than the gray, but reasonable alert dosimetry, you can get it down below 0.2 gray for the largest solar particle event we know. The cosmic rays, they're not very easily shielded. This is the total dose in gray, so the absorbed dose, that's what you want to think about when you're designing research, and this is the heavy ion dose. So the problem is most of the heavy ion research that's funded by NASA and NSBRI has used doses a half a gray or higher. So it's not relevant for tissue effects because tissue effects have thresholds, and if you're way above the threshold, you're not answering the question. The question should be, are you below or above the threshold? So to do that, you need to get down your research study down to these doses. There's a solar cycle effect. This shows you in different parts of the brain. There's not much change as you go through different parts of the body because of the shielding effect. The heavy ion dose is between 1 and 5 rads for annually, a little bit higher, maybe 2 to 8 rads on a Mars mission because it's a little bit longer than a year on the Mars surface. You don't get many heavy ions because the body of Mars cuts it down, the atmosphere cuts it down. So that would be the doses you want to know. So non-cancer risk, radiobiology teaches us that you have thresholds, and it's not just a threshold, you also have a probability that increases with dose above the threshold, but a severity. The severity could be severe clinical syndromes, but also a shorter latency from the time of exposure to the expression. So most of the studies are not looking at that. So I estimate over 95% of the studies in this area, non-cancer effects, are using doses way above what NASA's interested in, what would occur on a Mars mission. The other thing is dose rate. Dose rate's not important for high LET, especially at low doses, because you're only hitting particles once, but you do have protraction effects. And this is something for the central nervous system. Dr. Lamoli's going to talk about that. Is going to be very different. What is a protraction effect in the CNS? It could be very different than cancer, which there has been some studies. Dose rate effects are important for low LET radiation, like protons or helium particles, the high energy carbon particles, but protraction, nobody has studied that yet. The DOE low dose program insisted that everybody do below 0.1 gray. I think NASA should follow a similar approach. Okay, so the CNS risk, there's study in mice and rats show that there's differential results. So there are some important results below 0.1 gray, and I think Charlie's going to talk about that. But there's other studies that don't see these and the protraction effect. This is a model that I like, that you have an acute inflammatory response in the CNS that's protective and is good, but at some point it becomes chronic, and that's bad. And I think that's one of the main questions is, are the doses of relevant doses of a space mission going to be above or below where you're going to go from that acute protective inflammatory response to a chronic inflammatory response in the CNS. This is a model we did that was just published where we integrated all the data on hippocampal impaired neurogenesis and came up with a model of the doses. And you can see that for a Mars mission, changes to neurogenesis don't seem like they would be important for NASA. Other endpoints are just emerging, and we don't know if they're going to be important or not. More studies are needed. But that was one that NASA did a good job of focusing on. Are you going to have impairment of hippocampal neurogenesis, which is important for new memory? And I think it came to the conclusion that it won't be a problem. I'd like to see other endpoints in the CNS study to that same conclusion. Okay, so this is just so it showed you some data for performance testing in rats where 10 centigrade, which is 10 rads. 15, there is an effect. 5 rads, there's no significant effect. If you go lower, maybe no effect at all. Similar threshold doses here in rats. Now, other studies show effects in rats at 10 rads, so we don't know how that's going to pan out, and even a little bit lower, but a lot of studies show no effect. For protons, not much going on below 1 gray for behavioral testing of animals. Okay, this is that was just a plot that showed the energy deposition in the CNS. Circulatory, just one slide. The detriment is smaller than cancer. The life loss, is smaller. The excess relative risk per sievert from human studies is about 0.1 for circulatory diseases, and for cancer it's about 1, so it's about 10 times higher excess relative risk for some types of cancer, maybe a little bit lower for other types. the life loss is less. There seems to be a threshold for these effects, so you need to be looking at the relevant doses to see if it's going to be important for NASA. So the studies I've seen from the NSBRI and other people, they're using 1 gray of iron particles, no relevance for this endpoint. There's also a new study from the atomic bomb survivor, that's one of the main studies, which suggests that you would have to worry about circulatory diseases, where they're showing that there's a comorbidity with cancer, that there's issues only when you have the comorbidity. Okay, so the astronauts also, they have very low circulatory diseases, 3 times lower than the general population. The big question to me then is cancer. This is the only risk that's above the dose limits projected for a Mars mission. CNS, they don't really have limits, so we're not sure. There's major questions about the lack of data, and I'll focus on this non-targeted effect. So these would be the predictions for a Mars mission for the different types of cancer for a female of age 45. The risk of getting cancer could be higher than 16%. The risk of dying of cancer could be as high as 12%. You do have cardiovascular and ischemic heart disease in this. This is what's called the targeted effects model. The non-targeted effects model, assuming there's a bystander effect, we have a new paper coming out in Scientific Report, that could double the risk. So we don't know which model is the correct one. So the RBE with the non-targeted effect, the bystander effect, doubles the risk the first time it's been calculated. The other difference is that the quality of the tumors. Heavy ion tumors seem to be more aggressive. So this is an example. They metastasize at a higher rate compared to background or proton or gamma ray tumors, and they also give you different types of aggressive cancers compared to proton, X-ray, or background tumors. So there's something about the tumors produced by heavy ions that's different. So that's, to me, an issue. Mitigation, I think I'm almost out of time, but the issues would be chronic use. So what we know about mitigators is mostly for an acute exposure in a clinical setting. What do you do? What's known about chronic exposures? Model systems. This is data from Jerry Shea. His model can't look at low doses because you need to get a background below about 5% to do the doses you're interested in. So it's a big question. So end up the doses, again, too high, especially in the non-cancer studies. Cancer risk is above the risk limit for NASA. One question is, you hear it in the press several times recently, even last week, we could just waive the cancer risk limit. I think that's reasonable. The cancer risk limit is 3%. You could waive it for a 5% limit risk. So if the risk for Mars, all the research is done, is 5%, I think people would accept that. But what if it's 25%? Would you accept that? So you really need to know the answer before you say you would waive the limit. That's kind of a meaningless statement by NASA until they know what the actual number is.
Okay, thanks very much, Frank, and thanks for sticking to time. The second speaker will be Kathy Held.
Okay, so I was suggested that we talk about our background a little bit, so I'm going to skip that slide, so I don't think you need to do that since Gail's introduced me. And I do want to stress that since I'm with NCRP, what I'm going to say is not the views of NCRP or Mass General, Harvard. It's all mine to get myself in trouble. And as we all know, ionizing radiation background, the environment in space is quite different than what it is on Earth, and that's the challenge. And I've organized this talk to really address the 5 specific questions that Gail had sent to us previously. And I'm not going to show as much data. Frank has just shown you a tremendous amount of really impressive data. I'm going to be a little more hand-waving in what I say. And I want to start out with a general remark about the NASA Radiation Research Program, in my opinion. And I think this has been a very challenging and difficult program to develop because, as was talked about in the first lecture, there's a need for applied research to understand the risks. We got to have numbers to put into that risk model, which has been so elegantly developed and described by Frank and his colleagues. We've got to know quality factors, and we have to know dose and dose rate effectiveness factors to put them into that model to come up with numbers. I don't know how you get at that without just doing brute force kinds of experiments to get the numbers. And in particular, as this graph from one of Frank's studies shows, we need to know quality factors, we need to know dose rate effects. But we got to couple that with basic biology. mechanisms. What's the chemistry, biology, biochemistry that's going on? And without that information, we'll really never know how reasonable those numbers are when we extrapolate from animals, where most of them are going to come from, to humans. And without that basic biology, we're not going to know how to develop effective medical countermeasures. So there's a real dilemma here. They're not necessarily mutually exclusive, but it's somewhat difficult sometimes to describe and define a whole program that can do both of these things simultaneously. So maybe we'll do brute force and fill in the matrix, and to an extent, that's what's being done. People have been doing experiments funded by NASA with various ions in various animals and strains and species, males and females, looking at cancer induction, for example, in various tissues, different dose fractionation patterns, shielding. Is that going to get us where we need to go? Are we going to be able to fill in at a reasonable cost enough of those boxes to get the RBE numbers and the dose rate effectiveness numbers that we need to decrease the uncertainties that Frank has been talking about? How do we pick where to go? Or if we do enough of this, are we going to start to see some patterns that will become apparent that we can use from mechanistic studies? And we got to do mechanistic insights somewhere along the way as we do these brute force sorts of things, if that's the way we're going to go. We need to be able to have sufficient animals included to be able to study the various tissues for the mechanisms that we think are involved, for inflammation, oxidative stress, vascular damage, et cetera, to get the mechanistic information to be able to develop countermeasures. So one of the questions Gail asked us to address was, what are the major endpoints of importance for these studies? And Frank has already shown you very convincing data. Cancer is number one. We've known that for a long time, but also increasingly we're concerned about circulatory cardiovascular disease, and we're concerned about CNS function. So do we really have good information, good data to raise those concerns? Well, at high doses with photons in humans, yes. Much of the data comes from the atomic bomb survivors, for example, for cancer and cardiovascular. Some data comes from many other populations, including cancer therapy patients. In the low dose regime, we have some animal data for photon exposures. We have some space-like radiation, barely low enough radiation doses to really be relevant to the space system, as Frank has said. And when it comes to the low dose, less than 1/10 of a gray, in humans with the high LET radiation, there's nothing. So we really are operating on faith by extrapolating from photon data at high doses, mostly in animals and some in people, that these are the important in tissues. So we were asked to talk about mitigators. How much risk can be mitigated with what we know right now? In my opinion, the answer is very simple: nothing. Okay? Right now, we have some information on normal tissue responses that's been developed over the years from radiotherapy patients, from studies developed by the Homeland Security through the NIAID Radiation Countermeasures Program, to develop agents, medical approaches, chemicals, pharmaceuticals, nutraceuticals that can protect, mitigate, treat radiation damage to normal tissues from photons. We got zip when it comes to ionizing radiation, space-like ionizing radiation. And I think since most of what we have in photons doesn't work, I'm not sure that we know how to extrapolate to high LET radiation. So how much biology do we need? I could be flippant and I could say a tremendous amount. I'm going to try to be a little more positive and hope that there may actually be some underlying commonalities here for carcinogenesis, cardiovascular disease, central nervous system damage, in that all maybe include an important component of chronic oxidative or nitrogen reactive species stress, chronic inflammation. If that's the case, then perhaps we can ameliorate or mitigate a number of these detrimental endpoints by development of appropriate antioxidants and anti-inflammatory agents And certainly NASA should be looking at combining with NIAID radiation countermeasures program for some of the agents that they are developing to see if there are synergies potential there. What's the big damage? What are we worried about? Is it DNA damage? Is it reactive oxygen species? Well, we have to know this if we're going to mitigate any of that damage. From photons, we have lots of damage studies and DNA responses and so forth to suggest reactive oxygen species are important and that they produce DNA double-strand breaks that are important. From high LET radiation, we certainly know that the number of DNA double-strand breaks is high, but it's different kinds of damages. It's clustered damages compared to what we had produced by low LET radiation, where the reactive oxygen species are predominantly important. So, what's important? Well, it's going to depend on the endpoint you're looking at. Think about mechanisms. If it's cancer that we're most concerned about, then based on the normal beliefs of how cancer develops, there's got to be DNA damage. Presumably, double-strand breaks are important to initiate the response, but there's also lots of data to suggest that chronic stresses, chronic oxidative stress and inflammation in the microenvironment are also really critical and interact with the DNA damage. With cardiovascular damage, central nervous system damage, where there aren't so many critical proliferating cells, actively proliferating cells, then maybe the real problem is those reactive oxygen species, the chronic inflammation. Maybe that's what we need to figure out how to go after. And is that stimulated by DNA damage and the DNA damage response in any way that we don't understand? We need some novel thinking along those lines. So what do we have in terms of current mitigators? Well, classically, they're reactive oxygen species scavengers. There's also stimulation of growth factors and stem cells. There's also the anti-inflammatory agents. All of these have been around for many, many years. Again, we need some new ideas, and I don't know what they are. The last question was about dose being modeled, and Frank is really more the expert on that, and I think he's addressed it. I just want you to think about what's dose. By definition, absorbed dose is energy per unit mass, but what's the mass? When you have a track structure, such as just illustrated by Frank, what's the dose? Is it the dose along the column of that track? Is it the surrounding tissues in that? And this is just to illustrate that. Maybe it depends on whether you're looking at cancer or central nervous system or cardiovascular damage. And how do we figure out what's cumulative if we don't know what the dose is and what the stimulus is, whether it's DNA damage or whether it is some kind of chronic inflammation? Thank you.
Okay. Thanks, Kathy. I now want to move on to the third speaker, Charlie Lamoli.
Okay, so thanks for having me. So the last speakers gave really nice broad overviews, and I'm going to take a slightly different approach and try to point out some of some recent data from our lab and others of why I think NASA should be and should continue to be concerned about the effects of space radiation on the CNS. I think it's going to, in the end, be the single biggest problem NASA has to solve, not just going to Mars, but for extended travel beyond space. So you may have seen some press. The press gets a hold of some things and they kind of blow things out of proportion. People have called me up and say, Lamoli's going to go into space and get crazy and stuff. And, I don't know where they come up with these things, but it has certainly caught the imagination. I tried to set the record straight here by pointing out, I put an article out in Scientific American about dealing with some of the effects on cognition of space. And so let me take you through a couple of data sets to highlight why I think this is important. I clearly don't have the time to describe all the complex interactions of the CNS, but one thing that NASA has done a very nice job in funding is a lot of behavioral neuroscience, where there's a number of tests that we can give to rodent models that have been subjected to space-relevant doses of radiation of all types that interrogate different regions of the brain. And the result of some of these tests are frankly, if you'd have asked me 10 years ago, was very surprising. And so let me share with you some recent data now. This is unpublished data. This gives you a sense of where some of my work and my colleagues' work is going. This particular test here, called the elevated plus maze, is a measure of anxiety. And these measurements were now made 1 year after exposure to the proper doses, the low doses of helium ions. And what you can see compared to the control in blue, the percent of its entries into the open arms. The animals are afraid to go out into open space, which has been a measure of anxiety used by neuroscientists for years. So these animals are afraid, okay? Similar measurements made here with the strange test we call the forced swim test that's been used by behavioral neuroscience as a measure of depression. You put an animal in a beaker and simply look at how much time it floats versus the amount of time it tries to swim. When you give animals antidepressants, they struggle more. They try to swim. They don't give up. So this is a measure of despair, depression. So you can see here, the red and the green bars show that 1 year after exposure to the same doses, these animals are exhibiting classical signs of depression, okay? And so there's tons of behavioral tests that my colleagues and I can perform. These are some cartoons showing here some examples of spontaneous exploration tasks. And the key thing to remember here is in going from left to right, what we're really asking these rodents to do is recognize and discriminate novelty. So an animal can recognize a new location or a new object, and a smart animal will spend more time at novelty. So here's a series of studies that I find quite profound. The discrimination index you see on the left is simply a measure of the amount of time the animals spend exploring novelty. The blues are the controls. If you look at 6 weeks, 12 weeks, or 1 year after exposure, these animals aren't smart. They have significant impairments in learning and memory. This is shown by both the object in place task on the right, and in going from left to right from 6 to 52 weeks, this again is shown in a temporal order task, okay? So there's what appears to be a permanent decrement here after exposure to very low doses. I don't expect to go over this entirely right here, but this is what I consider some astounding data from Stanford now. And these animals were the same animals shown here. We sent these to Stanford, and what we're measuring here, the controls are in the white, and the black bars in A, C, and E are lower, and you can see there's no black bars on the right. We're stimulating a neuron in the hippocampus and asking whether that stimulation event can trigger a firing event in a region of the brain called the parahynal cortex. This measures long-term connectivity. At 6, 12, and 52 weeks, the connectivity between the cortex and the hippocampus is virtually abolished. This is a permanent change after exposure to helium, all right? So what I've shown you is that clearly cosmic radiations have a lasting and profound impact on the CNS, but why? And so this leads me into some other data that I'm particularly fond of, where we look at structure-function relationships in the brain. And one of the approach our group and my colleagues have looked at, they take animal models where the neurons are green, and this facilitates a morphometric analysis of these neurons. And what we've done over the years now, over the last 3 years specifically, is we've measured reductions in the complexity of these neurons. So going from left to right, we have an unradiated animal, and now we look at the brain after exposure to 10 centigrade or gray of protons, and we can quantify these morphometric changes by the bar chart you see. And what we can see here is after a month in this situation, the yellow bars show a marked decrease in the structural complexity of these neurons. If we go to a space-relevant dose, for example, of titanium or oxygen, don't get bamboozled by all the plots. What you can see compared to the red bars on the left is every single structural parameter shows that the neurons are compromised in terms of branch points length, suggesting that the complexity of the neurons is going down. Now, if we zoom in on these green plots here on the left and look at these little red structures, which are dendritic spines, these are what we call the structural correlates of learning and memory. They contain the synaptic machinery that mediates neurotransmission. If we look at the control at the top or the bottom panel, right, and the top panel would be 6 weeks after exposure, the bottom panel 15 weeks after exposure. You can see as we go from the control down to 30 centigrade or 5 centigrade, titanium or oxygen, there's less spines. So the dendritic spine density is lower. This isn't good, and this doesn't resolve. These changes, again, appear to be permanent. So the spine density, the things that mediate neurotransmission, radiation is dropping this. And so this is the last data slide here. Here we have some data here that Frank and Kathy have touched on about inflammation. At 15, 27, and 52 weeks after charged particle irradiation, we see significant increases in neuroinflammation. This chronic inflammation is playing a significant role in impairing neurotransmission in the brain. And so what I've shown you now is that I think some of the morphometric changes to neurons caused by radiation exposure underlie the cognitive deficits in the specific regions of the brain we look at that are interrogated by any number of behavioral tasks. Underlying this, I haven't shown much stuff. Many groups have shown significant alterations in synaptic proteins, major increases in neuroinflammation. So what does this mean for NASA? Why should NASA be concerned, or why should NASA continue to fund radiation work, particularly in the CNS from my perspective? Is it increases the likelihood of manifesting cognitive dysfunction. I don't think there's any doubt about this. Multiple types and doses of ionizing radiation have been found by at least a dozen of my colleagues now, where we get disruptions in learning and memory, cognitive flexibility, fear extinction, which is incredibly important. We can come to this later. Executive function, anxiety, and depression-like behavior. So the important thing, too, to point out for the CNS is this increases the risk, I think, for developing mission-critical performance decrements, okay? And it also obviously increases the risk for developing long-term neurocognitive complications. So these behavioral impairments, we believe, are caused by these alterations in neuronal structure, elevated inflammation, and in short, radiation exposure to the CNS is promoting the emergence of aberrant behavior, and we have got to get a handle on this. Precisely what these changes are going to vary between individuals, but over the long run, these are problems that aren't going away, and they're a serious threat to the long-term well-being of humans in space. Thank you.
I want to thank all three speakers for sticking to time very carefully. Maybe it was my threats that did that. But in any case, I want to at least spend a couple moments going back and trying to get the panel to talk about a few issues that maybe we could clear up a little bit. The first one was everybody sort of talked about mitigation a little bit, but nobody talked about mitigation and shielding. And Charlie, I didn't hear you talk about mitigation of CNS effects. So could I hear some comments about, first of all, the role of shielding and also possible mitigation with CNS?
Well, I mean, there's really two ways that we could mitigate any of these radiation effects. Obviously, shielding, more advancements in shielding are going to help everyone. As Frank pointed out, to my knowledge, there's no perfect shielding strategy for the high-energy particles from the GCR. For the brain, there's biologic countermeasures that I, along with other people in the CNS field, are working on. And really, a lot of these strategies are focused on, I think that the first focus should be minimizing the inflammation in the brain, and along with that, strategies to protect what we have. Because once the neurons are damaged, it becomes much more difficult to regenerate them So I think the approach should be, we got to protect the host neuronal circuitry as much as possible and preserve what we have. And that's the same type of approach that the neurodegenerative field and Alzheimer's research really takes. I mean, once you have 25% of your neurons gone and you're going from mild cognitive impairment to full-blown Alzheimer's, now I'm not saying radiation causes Alzheimer's, but the strategy for the clinicians is to protect what you have. And I think for NASA, shielding biologic countermeasures that protect the brain from some of these radiation-induced decrements really is the way to go. And that's really, quote, a no-brainer. So.
Yes, I'll just say something more about. So my first slide, we showed you, and it's well known that solar particle events can be shielded. You don't need, I think that was in the Gerstenmaier in the news last week about funding this thing. You don't need that because current spacecraft have enough shielding. You just need to know a solar particle event started. They last 8 hours to 2 days. Everyone's different. So there's enough shielding on spacecraft to get that dose down below 0.1 gray. You maybe wear that thing to get it a little bit lower. And then GCR, it's slowly absorbed. So you get some change in the first 20 grams of aluminum. That's like the average spacecraft thickness is about 20 with all the equipment. And then it doesn't really change after that. You can make things worse with the wrong materials. And so the neutron effect is what you're worried about, that you produce neutrons from the shielding. So if you use too heavy of a material, you get a lot of neutrons. So you want to keep that as low as possible. It's an interesting question for cancer. It might be different than CNS, because the CNS, if it's a heavy ion problem, maybe shielding is the answer. But for cancer, you're converting the heavy ions into lower particles, and the lower particles seem to have a lot of cancer risk, almost as much as the heavy particles. So the stopping protons that you make from neutrons are just as bad as the heavy ions. So we don't know about that. For the CNS, it could be where shielding has a different role than it does for cancer risk.
One other question I'd like to ask, and then we'll open it up for everybody to ask questions, is it seems like there's a bit of not so much a disagreement, but maybe the role of CNS and cardiovascular disease were not played up so well in the original decadal study because we didn't know as much. And I guess I'd like to come back to that question of cancer versus CNS, and it sounds like, Frank, you're sort of arguing that the cardiovascular risk may not be so significant, but I'd like to kind of go back to that and talk a little bit about where are the most important risks at this point in time.
Okay, well, I think to me it's not an either or. So cancer is you're above the limit for a Mars mission unless you go at solar max. So that's a big risk, and it could be that we call it the conventional model, the model that was reviewed by the Academy of Sciences in 2012-13. You're above the limit, but that doesn't have non-targeted effects in it, and it doesn't have that the lethality of the high LET tumors might be worse. That could push the risk up above 20%. So those are the really big questions. Now the cognitive changes, that's important too, but there you're kind of stuck because you only have mice and rats. And Charlie has a consistent story, but if you look across investigators, there's differences across investigators, and that's partly due to the different types of radiation, different particles being used. So maybe it's more consistent than we think, but no one's addressed a chronic exposure yet. So there's a lot more work to be done in the CNS. I think cancer is more focused. You need to answer that non-targeted effects question. You need to answer the question about the lethality of the tumors. If you could say they're both off the table, then it's not so bad.
So I agree with Frank. I don't think it's an either or. I think given the preponderance of what we know much more about secondary radiogenic cancers from all types of clinical and environmental exposures that have certainly informed us about what we might expect from the different radiation types. I still think the 800-pound gorilla in the cage is the normal tissue complications in the late responding tissues. So because we know much less, and we're never going to know as much as we do with the cancer. We just don't have that in. But I think the normal tissue complications in the CNS are for me the critical question to really to focus on, without compromising the other work along. There was just so much we don't know about that at a mechanistic level, and we're working on translating the rodent findings to the human study scale. But there is also no data that I'm aware of that suggests that a rodent neuron will respond any differently than a human neuron at a biochemical cellular level. So the changes that we see in rodents, I'm also do other clinically relevant, not NASA relevant studies. These are what's occurring in our patients subjected to cranial radiotherapy. And we know that these patients, we've known for decades, develop a progressive dementia that does not resolve and gets worse and worse. And so this gives us some expectation of what might happen at much lower doses, but with very different radiation types. And the impact that the CNS effects might have during the mission is the additional concern, because you're not going to get a cancer in a year in space.
Go ahead, Kathy.
I just wanted to amplify essentially what Frank has been saying, has said. And I think we have a lot more human data, albeit mostly photons, low LET, from many different epidemiological studies over the years to support the importance of cancer than we do for the other endpoints. So the human data is starting to show us the long-term epidemiology data, increased cardiovascular damage. We've got nothing except the high-dose radiation photon data from patients for CNS. And that's really a big decrement, a big hole in our knowledge, is to know how to extrapolate the wonderful data that Charlie has shown you for the animal effects to humans in space-like relevant radiation doses, not the much higher clinical doses.
Okay, thank you. Panelists, we'll now open this up for anybody here that wants to ask a question. I'll remind people, if you want to ask a question, give your name and what your institution or what your affiliation is.
Yes.
Please.
Okay. My name is Chuck Fuller, University of California, Davis. Gravitational physiologist, animal scientist, neuroscientist background. I was curious about kind of two or three different things. One, the word biomarker never came up, and I'm wondering if there's any opportunity in terms of particularly some of the inflammatory, ROS, other things, whether there's a potential of biomarkers that might be a useful tool. Two, the lack of mentioning of the immune system. I would have expected the immune functions to play a more visible role in the discussion of some of the outcomes that you're talking about with radiation. Again, I'm not a radiation biologist. And number three, this kind of couples back the strong discussion on the CNS. CNS is a big organ system, has a lot of functions, and most of the discussion I've heard so far has been on behavior, neurocognitive type of effects, and there are a lot of other areas of the nervous system in terms of regulation, neuroimmune, neuroendocrine, and I'm just curious whether or not these have not simply been looked at, or whether or not the effects are not anticipated to be displayed in those functions of the central nervous system, which, if you have neural damage in general, stochastically, I would imagine some of these other subsystems would also have sensitivities, hypothalamic systems and regulatory systems and so forth.
So this is a question that could probably be answered for a whole symposia, but I'll ask the panelists to be very short.
So I'll mention some. So the biomarkers, remember we were given 10 minutes.
Yeah, I know.
So the biomarkers has not escaped the attention of NASA or the NIH, and people are working on this. As you can imagine, any types of omics studies looking at serum or CSF or whatever tissue-specific biomarkers needs to be vetted out. Because the radiation damage whole body at that level is aspecific, the biomarker question is much more complicated for radiation than perhaps other areas. The role of the immune system is certainly important. Let's just, for the CNS, the microglia or the innate immune cells, they go up after a year. If we get rid of the microglia by inhibiting CSF1, we can make the animal smarter. So there's a mitigating agent. So getting rid of the microglia after radiation exposure reverses the cognitive effects. The role of the peripheral immune system, at least in the CNS, is much less important, largely because the doses are so low. And changes in the, hematopoietic cell counts that would obviously impact the immune system after low-dose radiation in space. They're kind of like all over the place. I don't think they're important at all.
Yeah, so the biomarker, I think I'm a member of NCRP, and Kathy's one of the officers, the vice president. But in radiation protection, you don't send people if you're above the acceptable risk level, or you would infer clinically significant effects. So when I think about biomarkers in astronauts, I think, well, then it's too late, right? After they're exposed. So I think there's a role for it, but we're kind of more on the other side. Is the risk acceptable? Are you going to have clinically significant? So I think that the biomarker area is underplayed at this point, but once you have a mission, I think there's a more important role.
Just to elaborate on that a little bit, I think Frank's right. Practically for the astronauts on a mission, biomarkers are probably too late. But in basic biological experiments and trying to understand what's going on, to get biomarkers or surrogates endpoints for some of these very late responses that we're trying to look at for the animal studies would be marvelous if we had them. Is that we knew they were true surrogates or biomarkers or indications of, a heart attack years later. We don't have them, but that would be really fruitful to know.
I don't think we answered the immune system issue, but the only comment I'd make is that there are a lot of NASA investigators that are looking at immune modulation following radiation responses, and I think some of Charlie's work that he presented actually was with inflammatory responses that are immune mediated.
Okay, Jeff Smith. I'm with the ASGSR Society and also NASA Kennedy Space Center. I think that two of you pointed out the lack of experimental results for using long-term, very low-dose, high-energy radiation. And I don't think that's from a lack of interest on your part or NASA's part or on the scientific community's part. I feel that's due to the fact that the NASA Space Radiation Laboratory really, the NASA-sponsored facility for being able to conduct a lot of these types of experiments just can't practically perform those kinds of experiments for us, and so therefore, over the past number of years, no PI has been able to really propose to perform those studies. And I wonder, is that a critical gap or not? Am I misunderstanding? And if it is an important gap, what could be done about it?
I'm actually chair of the committee at Brookhaven called SACR, Scientific Advisory Committee for Radiation Research, that oversees beam time applications for that use of that NASA facility. The facility is great. I'm obviously prejudiced, but it's very, very useful. It could do many of the experiments that we need if there was enough money to do it. It boils down, in my opinion, to money. In many cases, it's not a problem with the facility. There are some limitations to some of the things, and I don't know what you specifically have in mind. But for instance, that matrix I was showing, many, many of those blanks could be filled in if we had the money to do it.
Right. I mean, as Kathy pointed out, and as Frank is well aware, we've just getting the capability to more faithfully reproduce the galactic cosmic rays by adding multiple ions. I mean, 10 years ago, it was hard enough to get any given ion, any given dose, and so now our capabilities give us the ability to simulate the GCR. Now, whether we can simulate the GCR over a 2-month exposure, sure, if we had enough money, but we just heard our budget's flat. I mean, so a lot of what we do is, tethered by practicality. And that's just...
Yeah, so Kathy has done very low dose, like 10 times lower than the space doses, so that's possible. In low dose rate, we implemented a large beam, so much larger than the typical beam size. Cancer studies have been done over like 3 or 4 weeks by just doing a small daily fraction. And for cancer, I think that's reasonable. But for CNS, I think that might create some issues about whether that's realistic to bring an animal in and out. So I think people need to work out how you would do a chronic CNS exposure over like a Mars mission in a mouse is like a month. duration. So you'd want to do a month duration exposure. How you would do that would be some practical things that would have to be developed.
It's just practical. It could be done, in my opinion. I'm not an animal person. Charlie's really the animal person, but I don't see why, if the will was there to do it, that couldn't be done at the NSRL facility.
Thank you. Yeah, how important is it to have that will? That's what I was getting at. Thank you.
Thank you. Rob. Oh, yeah. I'm sorry.
I'm Marty Glicksman. I'm Dean of Engineering at Florida Institute of Technology. I'm not a life scientist, but what immediately comes to mind listening to the panel of experts is sort of reminiscent of the discovery some years ago that the vascularization of solid tumors in rodents could be suppressed by certain substances derived from the tumors themselves. I forget the famous researcher who came up with this. And when asked, does this portend basically a cure to cancer, he said, no, I'd rather put it that this is some very good news for rats. And I'm hearing a lot of rodent testing, for ethical reasons, obviously, but maybe it's just good news for rats or bad news for rats.
Well, I think Michael Fry had done a lot of good work showing. So, you know, you sort of is a mouse a good model for cancer risk in a human? So you have the similar DNA damage structures, DNA damage repair systems. You have an inflammatory and immune response. They're all there. But he also did some studies where you compared relative risk factors for gamma rays between mice and the atomic bomb survivors, and it's a very good correlation. So I think, you know, for mitigation, that's different, but for just assessing the risk, coming up with relative risk factors, I think it is a very good model.
I'd also like to point out, we hear this constantly about how do you know what a mouse does is what a human's going to do in a spacecraft, and we don't exactly, right? And we never will exactly. But there are a significant number of cognitive tasks, for example, a sequence odor recognition task, where you can give to people and rodents, and the people make exactly the same mistakes that the rodents do. Trust me, the data's out there suggesting that everything we do in rodents actually does have applicability to humans. I've looked at it extensively. Not everything, but when you, yeah, but when you have 15 researchers giving all their different tasks and they're all finding decrements, it is suggesting that there could be concern to humans, although they may not want to push a bar up and down. There are tests out there that show remarkable parallels that give us confidence that what we're doing is not just a rat-happy experiment. So.
Yeah, the other anecdotal information is that many of the space station astronauts come back and go into management. What's going on?
As a dean, I understand that.
Rob.
Rob Furrow from the committee. First thing I want to do is remind the people that are listening online, if you want to email a question to the group, you can do so at microgravity midterm 2016 at NASA, at nas.edu, and we'll try very hard to get your question before the panel. My question for the panel tugs on something Catherine said, that as a genomics person really catches my interest. And that is, you said high LET causes a different kind of DNA damage. I'd like to hear a little bit more about that, especially in the whole genome sequencing era. So can you to broaden the question a little bit, can you talk a little bit about what our knowledge is of DNA rearrangements that occur at cosmic space flight exposures that we know now in the genomics era that we didn't know back when we were simply looking for mutations?
You guys might have to help me out. based on sequencing, genomics kind of data, we know that there are different gene expression panels, different at high LET versus photon low LET radiation, high doses versus low doses. We know that there are different, more complex rearrangements of the genome that we can see even at chromosome aberration levels, which we've known for many years after the high LET radiation. To go back to where you started with, what I was talking about in terms of different DNA damage, is that we know that the damage is much more complex, or so-called clustered damage from high LET radiations, because of those track structures that Frank showed you so elegantly. When those go through a DNA molecule, they put a huge hole in the DNA, many, many ionizations in a very close proximity, that it is much more difficult for the cell to repair using the DNA repair machinery properly and accurately, compared to what occurs with a photon exposure, where there are more isolated single ionizations or small clusters of ionizations that might produce more isolated DNA damages that are more readily repaired. So and there's the problem with those. There's a problem here in that we have difficulty measuring and really understanding in quite the detail we want those clustered lesions. What we can measure, for instance, is surrogates. How big are the clusters of repair proteins around the clustered lesions? And they're much bigger clusters around the high LET-induced clustered lesions. Having physical measurements of that damage is a little bit more difficult because the assays we have all show up as double-strand breaks, because it collapses to that when you got a really complex lesion. So there are clearly different DNA damages that either have to be repaired differently, stimulate different DNA repair pathways to some degree, preferentially, and result in different DNA lesions, different gene expression, and endpoints like that.
Gotcha.
Yeah, I agree with all that. And then one study that I think that was impacted by budget cuts at NASA, human research, was Mike Cornforth at UTMB. He's a world-renowned cytogeneticist, and he's always telling you when you have a chromosome aberration, you make a small ring, and he claims he can see the smallest rings. So, 5 megabase pairs, and he was funded to do the full genome sequences to see, that's one of the open questions, these cluster damages, do they lead to very small deletions from the non-homologous end joining repair pathway? And nobody really knows. So he was funded, but that because of the budget cuts, he didn't continue that study. But there's lots of studies that you could do with more funding.
And just want to point out that the difference between these radiation types has been known. I mean, NASA knew this before they funded this stuff, and it's the radiation chemistry is linear with dose. Don't let anyone tell you otherwise. And so we know the initial yields of damage is the response to the damage to the high LET that will vary. And really what we're studying with the space radiation effects is the delayed repair of double-strand breaks, because they're more complex. And that's where the biologic response starts to tease apart the differences between a clinical dose of radiation and something you might get on the way to Mars.
Thank you.
Jonah Peter, ASGSR, over here. So we saw some pretty convincing data about induced depressive-like behaviors and reduced dendritic arborization. I was wondering if there's any evidence for elevated glucocorticoid activity or reduced BDNF levels, and if so, could exogenous neurotrophic factors be used as a potential countermeasure?
So the evidence for those changes after clinical doses is undeniable. The evidence after space radiation doses is non-existent. In fact, we've looked for BDNF changes after those changes, because obviously that would clearly impact a number of the endpoints that were shown. Nothing too significant yet. But that's not to say that neurotrophic supplementation wouldn't be a viable mitigator. So, I mean, that certainly is an area of some potential interest in research. But remember that at 5 centigrade, a lot of the things you might find in the literature, in a cancer research article, for example, are occurring after, the equivalent of, 60 gray. And we're not even in the ballpark of that total dose.
We're almost at our end point. I'm going to call for last question now. April?
April Ronca from ASGSR's representative and also NASA Ames Research Center. And I just wanted to bring up the topic of cross-cutting factors, some of which were raised and some of which were highlighted in the decadal survey. Dr. Kusanada talked about effects on women. I think that the sex differences and gender difference effects are critical to keep in our investigative lens. In 2012, there was a series of papers looking at sex and gender effects on a variety of physiological systems, and really what it identified is so many things that we don't know. I think it's important to keep that in our view, as well as aging. Dr. Lamoli brought up effects of recovery, et cetera, and I think that lifespan studies as an approach in our basic research is extremely important for us to really understand the scope of these effects. And then a third one that I didn't hear anything about, and maybe the panel wants to comment, is microgravity by radiation interactions, which can be additive, and there's now a growing field here, and how can we tackle those kinds of questions in this time of a flat and declining budget? Thank you.
So Ruth has been looking at microgravity and radiation effects on the bone, and there's some impact on the trabecular bone structure. But in the end, I think that even she would argue that most of the changes in her Heinlein unloading model have been due to the proton doses that she used. But I do think moving some of those types of studies, looking at some other types of normal tissue injury, cardiovascular disease, CNS would be of interest. I think the impact on age is critical to the extent that NASA might vary the age of the astronauts they send up. And your point on sex differences is critical. And I can share with you some data that we have, and I know that's been backed up by some elegant studies by Cindy Lemire at Harvard, and that females are resistant to radiation-induced cognitive dysfunction. So NASA should send up females. They're not showing the same. So there's something to think about.
But on the other hand, they're more sensitive to cancer induction.
Okay, well, I would like to thank the panel for their tremendous work, sticking to time and giving us, I think, a really amazing overview of the field. Thank you so much.
Okay, the break is out in the hallway here. The restrooms are across the hall. We're going to reconvene sharply at 10:30, so please be aware of that. The last thing I would like to do is, students in the audience, could you raise your hand? Yeah, so this is just awesome that you folks get a chance to see high-level science and policy discussion in action. Please do feel free to continue asking questions. Enjoy the break. We'll see you back here at 10:30.
I'm Elliot Meyerowitz from Caltech, a member of the committee, and here to introduce the Biological Adaptation to Spaceflight session, our second session of the day. Although I would say biological adaptations to spaceflight, had I labeled it. That is, to put it in Gail Allen's words, life support and habitation on long-term missions, such as missions to Mars. There are three aspects of this, and I think we'll hear about all of them today. Human adaptation and the animal experiments that test human adaptations to microgravity, to confined environments, to the bacterial environment of a spaceship that involve physiological issues. The psychological issues will be session 6. There are questions of bioregenerative life support, that is plants for micronutrients. A vitamin pill doesn't last 4 years. There's no better way to keep the nutrition of the astronauts appropriate, even if plants are not providing the predominant source of their calories or protein, but to provide the micronutrients, there's no better way than to grow some seeds and eat a little bit of salad. Or for water for purification or oxygen regeneration, and bacterial and fungal adaptation. The fungi that infect plants on the International Space Station are things that aren't ordinarily pathogens on Earth. We have to understand the adaptations not only of the plants, but of the microorganisms, and the same goes for potentially for diseases, bacterial, viral, and fungal, that the astronauts might suffer in space. What are the differences in the space environment, and what sort of adaptations have those organisms taken so that we can monitor the environment in ways that are relevant for the health of the astronauts? Our speakers are Charles Fuller from the University of California, Davis, a specialist in regulation of circadian rhythms and sleep, homeostatic systems, energetics, and metabolism. Cheryl Nicholson from Arizona State University, effects of biomechanical forces on living cells, both human and bacterial. And Annalisa Paul of the University of Florida, president-elect of the ASGSR, and maybe president by now? Not yet. Regulation of plant gene expression in response to extreme environments, including epigenetic changes. Charles, it's all yours.
Thanks. Oh, okay. Well, when Elliot invited me to present, he left me with a couple of rhetorical questions and a 500 or so page document to say, where are we? Considering a career in gravitational physiology and trying to say something in 10 minutes, the challenge was daunting, to say the least. And so I'm taking a different tact than many of the prior speakers so far, although we're just getting started today. I have no data to show you. I have lots of data to talk about if anybody has questions, but the issues are, where are we with what was originally recommended by the decadal? What can we talk about? What can we say? Where are we progressing or not? Where are we stalled? And I've tried to pick out some highlights to talk about some issues that I think are important and relevant that we haven't moved forward on enough yet, and other areas which are tied to the agency, its structure, its management, and the needs and desires of the science community as a whole. I was asked to say a little bit about myself, as in the prior session. I've been a researcher in physiology for a long time in this area, neuroscience. We run a facility where we use ground-based centrifugation facilities as one of our models. The other areas, I've had a lot of experience with a variety of models, from fungus to non-human primates with various flight experiments in space. So I've built flight hardware, run flight experiments. And with this background, I wanted to pick on the following few topics in my window of time, and then we'll be happy to answer questions afterwards. First of all, and I think we talked about this with Gail, NASA is struggling like other agencies that are mission-directed. It's a mission-directed agency. We talk about spaceflight, we talk about exploration, and the question of where does research fit into this model. We have applied research. What do we need to know about the crew and the environment and maintaining that environment to house the crew for long windows of time? Whenever we have living specimens on board a spacecraft, there are unique challenges to understand. And the longer that stimulus is, the longer that duration of that mission, the greater those challenges are, because there are undiscovered frontiers, such as the current visual problems that have cropped up in the human research program with the long duration exposure of the astronauts to spaceflight. The underlying etiology of that is still not understood. I think we're starting to gain some insights, but it's a new issue. The interaction between radiation, long duration exposure, between aging, gender, and so forth, again, are all issues that have been touched upon in the last session. We also have a structural organization within the agency in dealing with these issues, in which, when we look at life sciences as a discipline, which is how I would look at it from the outside as a researcher, we don't have a single integrated program. We have Space Life and Physical Sciences program, which does the fundamental space biology. We have the Human Research program under a different structure of management, which is fairly species specific in terms of the flight of astronauts and the development of countermeasures. And it's an applied program to deal with the issues of concern. We have the INSS Science Office, which actually is very platform specific by its definition, and it also has great influence on science and science funding. And then we have CASIS, which actually is also doing science, but it's of a commercial nature. It's directed under a different mission. It's not subject to the decadal call, but at the same time, it's very platform specific in the sense that it basically owns and controls 50% of ISS resources. So it's the interaction of these that has to lead to the integration of a program. The overall research budget, as was already identified by Gail and most of us know already, was limited, and we also have relative time limitations, particularly in a flexible scenario where we changed administrations recently. We've changed the structure potentially when we're going to be flying humans and what the needs for those early exploration class missions are going to be with humans. So that leads to changes in programs that are ongoing. So in summary, while we have an inter-program, interagency collaborations are just beginning, and this was a high priority call in the decadal, we're not there yet. We have been working on it, but it does not exist in terms of this interim status. We also talked about push-pull. I would have talked about applied versus basic research in terms of humans and the needs and the understanding of the role of gravity in living systems. We need to have ground programs as well as flight programs. There's a continual push to talk about what have we done in space, and we need to understand that we can do a lot of this on the ground. We don't have the stability of funding. We have the priority going towards the space programs. Ground programs are a lot less expensive. We can do a lot with them, and we can help develop the understanding of gravity using ground programs of various kinds and with various models. I picked 3 topic areas that I think are important to keep in the back of our minds. The one that Dr. Ronka mentioned at the last session in the Q&A, sex matters. Biological sex and gender differences and G-sensitivity are an important issue. We know they exist. We do not understand it well in terms of the astronauts. We have not had enough women in space for a long enough period of time, and we have not developed the specific data collection and epidemiological research necessary to understand what those differences might be. However, we know from animals that males and female mice, rats, significant differences in a variety of areas. And we've got a substantial amount of documentation on that. And so a lot of this can be done in animal work, and I think a lot of the folks that do clinical human applied research think, well, gee, we've got an estrus animal versus a menstrual human. They're not the same. Well, again, I would argue, based on the same type of discussion we had in the last panel, that there are parallels. There are issues that we can extract from the animal research that would have general applicability to humans, or what we would want to understand better in humans in terms of sex differences. Size matters. Okay. Biological sensitivity to gravity is very much
in many areas of physiology a relationship to the mass of the organism. Humans are the largest biological mass that we're going to fly. Okay, and we don't have many surrogates. We've had non-human primates in the past. I don't think the agency is going to be prepared to deal with those models again. So I think the largest we have, is, again, the mammalian systems at least, is going to be rodents, and that would be a rat as opposed to a mouse. Mice are extremely important biological models, but I think the absence of our ability to fly rats right now is a critical loss. It was called out in the decadal. ISS Planning Office, I was told 2 weeks ago, is not planning on expanding what was originally designed in terms of the rodent research habitat in its original science requirements documents to house mice and rats. They're not planning on expanding to rats. I know there are some discussions that this may change, but that's the current status right now, and based on where we are with the decadal, we are not there yet. Centrifugation. We need centrifuges. We need to be able to do it both on the ground as well as in flight. What we can do with a centrifuge in flight is we can study between 0 and 1G. We can load the organism, whether it's on the ground or in space. Loading the organism is critical as a ground as well as flight model because it does one thing, particularly in animals. It loads the vestibular epithelium of the inner ear. Ground models, bed rest, suspension, whatever, inactivity, unloading, does not unload the primary sensory apparatus that the body utilizes to measure gravity, and that is the vestibular epithelium. And when you don't unload that system, it has a huge impact on what you're measuring. Okay, we can take it out and we can show these differences, and we also know, again, there are biological and sex differences. So I'm being told I'm done, and on my last slide, the absence of a truly integrated, well-funded, established program which investigators can count on, and this came up a couple of times already, means that we cannot train under ethical, I won't train a graduate student for NASA right now under any ethical basis because there's no future for those students, and I can't even guarantee that I would have the longevity of funding necessary to support a student for the duration of a graduate program. And I think one of the things we're missing right now, and this has been a threat for the last decade, is our inability to get a well-established program for moving students forward. And I don't want to discourage any of the students that are in this room. I am glad you're here. I agree with that. I applaud that. However, make sure you are qualified to get a job in a standard field and learn the tools to do research in space and how to ask the right questions, but don't rely on NASA. And I think I'm done.
Thank you. Cheryl Nickerson.
That's a bit, yeah.
Good morning. Wherever humans go, microbes go. The two are inextricably linked. You can't separate them. And that's very, very important because microbes are critical regulators in the transition between health and disease, a variety of diseases. It's also very important because microbes change very dynamically and very profoundly and very quickly, especially bacteria, to their changing environment. And when they do so, they profoundly change almost all of their characteristics, which impacts whomever they're in or on. So it's not surprising then that NASA's NASA's microbiological research platform is designed to cover a wide range of different disciplines as it pertains to human health during exploration missions. And today I'm going to specifically address one of those important areas, which is the risk for infectious disease. And in so doing, I will be addressing some of the committee's decadal recommendations from their last meeting. So here's our ISS, right? And it's a complex environment. It's really much more like a dorm room than it is a Starship Enterprise, all right? And that impacts the microbiology approaches because it impacts a variety of spaceflight operations are just dedicated to microbiology. Why? Because humans built it and humans live in it. And this is what you have. You have a lot of microbes up there. So you have microbial contamination of panels, including communication panels. You have them in clogging water systems with biofouling, and they can make people sick. All right, so that's why NASA has a robust microbiological monitoring program that they carry out on the ISS. So almost over the past 2 decades now, my lab has used both the spaceflight and the spaceflight analog platform to provide novel insights into infectious disease mechanisms that we cannot observe down here on the ground using traditional conditions. I will give a call out to that rotating wall vessel bioreactor down there in the middle of the right hand, right there, because that is one of our ground-based analog systems. Regardless of whether we've flown microbial pathogens or human cells, we have seen that they exhibit biomedically relevant phenotypes that they don't down here, and that's important for astronaut infectious disease risk, but it's also equally important for translational activities back down here, wherein we're working to develop novel vaccines and therapeutics to keep us healthy. So this one slide is going to condense 10 years worth of research in my lab, and I'm going to kind of try and go at it at a rather brisk clip. But our short duration studies using both spaceflight and spaceflight analog bioreactors with the major foodborne model pathogen Salmonella have revealed very interesting results in terms of how this pathogen responds to spaceflight. So first of all, spaceflight globally increases the virulence of this pathogen. We've shown this on 2 independent spaceflight experiments where Salmonella's virulence was increased. It took a smaller dose. We call it lethal systemic infections in mice. Likewise, spaceflight increased the stress resistance of this pathogen, so it's better able to resist being killed by stresses that your body normally throws at it, and it uniquely forms biofilms in flight, where the same pathogen under identical ground conditions couldn't do it. Likewise, we globally changed the gene expression in this pathogen. Hundreds of different genes were regulated very differently than they are in the ground. That included virulence genes, and we were able to identify a master response regulator of those genes, a protein called HFQ. Get back to that shortly. We were also able to come up with a way to turn off the increased virulence that we'd seen in flight with Salmonella by simply adding ions to the media in which we were culturing them. And we further showed that one of those ions, phosphate, appears to play a big role in allowing Salmonella to escape some of the defenses that your body throws at it. And then back to HFQ. HFQ is a highly conserved protein. It's in bacteria all the way up to us. And we were able to show that HFQ is an evolutionarily conserved response regulator to the spaceflight environment because it's not just Salmonella that uses HFQ to control its spaceflight responses. A variety of very different pathogens do that as well, including Pseudomonas aeruginosa and a methicillin-resistant strain of Staphylococcus aureus, and also Candida albicans. Now, recent work in Jamie Foster's lab at the University of Florida has shown that it's not just pathogens, those bad bacteria that use HFQ, but good bacteria, commensals, that can live with a host, in this case a squid, also use HFQ to regulate their responses to spaceflight, having implications for the microbiome. We've also flown 3 additional spaceflight experiments that we're working on data from now. The first one, STL Immune, was the first study to actually infect human cells in flight with a pathogen. We used human intestinal cells. We infected them with Salmonella. We also had the first ever study called RASIV, where we looked at the ability of spaceflight to enhance the protective immunogenicity ability of a vaccine, live attenuated Salmonella vaccine strain. And our most recent flight experiment was Micro 5, also called Phoenix, wherein this was the first experiment to actually look at the virulence in real time of infecting a living human organism, not human, infecting a living host with a pathogen in flight, and that data is being calculated as we are speaking. We measured virulence in real time. Just a quick call out to a couple of those experiments on STL Immune. This is infecting human epithelial, human intestinal cells in flight with Salmonella. We have now analyzed that data and shown that in flight, those human intestinal cells are showing a very exacerbated infection response. They are heightened in their infection responses compared to those same cells that we cultured on the ground, which is consistent with Salmonella being more virulent in animals. We also showed that Salmonella can invade those cells in flight, and it causes morphological damage to those cells in flight. Our recent Phoenix Micro 5 experiment was actually one of the most complex biological experiments that's ever flown. In real time, we profiled the start to the finish of an infection and disease experiment by using a little nematode. It has an intestinal tract from one end to the other, much like ours. And we, of course, used our friend Salmonella again. We have sorted through over 10,000 videos. We're through about 7,000 of them now, measuring the virulence in real time. We are doing, for the first time ever, the transcriptome profiling of both the host and the pathogen. So you can see dynamically what both of them are doing. And and we tested a countermeasure to prevent infection from happening in the first place. Our two new experiments we have are in line with decadal recommendations. First of all of the studies that everyone has done so far on microorganisms have involved short-term cultivation. We've got to go to long-term cultivation. We're going into deep space and exploration missions. So we will have the first experiment that will study the effects of long-duration culture of salmonella and multi-generation growth in the chronic stress of microgravity. Are we going to see the same thing we see with short duration? We don't know, but we have to find out. It's going to be an extensive study. We're going to profile a lot of things. At the omics level, we'll be performing the entire genotype from both mutations occurring in the DNA to epigenetic changes at the methylome level. We'll be performing global transcriptomic and proteomic studies, as well as phenotypic studies of antibiotic resistance, biofilm formation, and pathogenesis-related stress responses. Mechanistically, we want to understand the mechanism, so we're doing a wild type and an HFQ mutant strain. And one of our other recent experiments with Bob McLean is the PI. It's great to study individual responses of microbes alone, but it's critical we study polymicrobial responses, which is another guideline or recommendation of the decadal panel, because microbes like to get together and form these sticky, gooey biofilms, which typically aren't good for things in flight. Right? They clog filters, they corrode systems, and they can cause disease. So in collaboration with Bob, we're flying two microbes, looking at together, mixing them, looking at their ability to form biofilms on ISS materials, including stainless steel, looking to see how easily those biofilms are eradicated with the type of disinfectants they have on orbit right now. And then also, we're going to see if those biofilms corrode that ISS material. This, of course, is critical to ECLIS, environmental control, and life support system design. And then recently, just last week, so the timing for this is great, the Alfred P. Sloan Foundation, in collaboration with NASA, just came out with their first postdoctoral award fellowships to study space station microbes. This was a part of their program for the studying the built environment, built microbiome of the built environment, and delighted to say that my postdoc was fortunate to receive one of those scholarships. So she'll be looking at polymicrobial biofilm formation using actual bacteria that were isolated from the ISS water system to see how they form biomaterials on biofilms on ISS materials and how easily they can be controlled. Real quickly, I'd like to cover some notable studies, many from other labs as well. With 10 minutes, it's hard, so I'm going to give a shout out here to other critical studies that are following guidelines of the committee. Cynthia Collins did some lovely work following up on Bob McLean's initial discovery that Pseudomonas aeruginosa can form biofilms in flight. She's shown that they can form novel architectures in flight, which could have implications for clearing them. I've got you thinking for that time. Jamie Foster's group continues to do wonderful work in studying the host microbiome interaction with the squid and mutualistic good bacteria, showing HFQ and transcriptomic changes related to that regulation. Dwayne Pearson's work, continuing pioneering studies of showing latent viral reactivations in astronauts, which is a direct indication of immune system dysfunction. He's done this for short duration work, but they're about to have a paper come out in NPJ Microgravity where for the first time long duration studies, guess what? Latent viral reactivations for the entire 6 months for those astronauts. Crystal Jang at Lawrence Livermore doing nice work developing microarrays to study different types of pathogens that could be present on the ISS. Craig Venter with Hernan Lorenzi, Mark Ott, and Dwayne Pearson, who, along with independently Fred Turek's lab at Northwestern, which are studying the astronaut microbiome, including the recent twin study. Paper that just came out in NPJ Microgravity by our team was the first transcriptomic profile of changes occurring in the whole blood of astronauts in response to spaceflight. We were able to show changes in genes encoding DNA repair, oxidative stress, and protein folding and degradation, and several of these genes are involved with viral reactivation and proper immune function. And then finally, wrapping up, I just want to give a big shout out between the profound synergistic alignment with about every function of our body, not just infectious disease, but immune system function, metabolism, behavioral disorders, and a number of these kind of properties we have to be concerned about, including cancer. Infectious diseases cause about a third of all cancers. And critical work by Brian Crucian's lab, which also just came out, has profiled immune dysfunction in astronauts. He's done this for years, and short duration, we know their immune system is impaired. Well, now guess what? Long duration studies, their immune system is equally dysfunctional. Okay, so it's extending into 6-month time limits. And he collected blood in flight, too. So it wasn't just before and after. And then in a big shout out, because we have to know as we go into deep space, what is our health in terms of the astronaut health? What is the health of the vehicle in which is carrying them? You've got to continue these kinds of monitorings and profile. How do you do that when right now the samples come back down to Earth? Well, how about sending up your first female microbiologist astronaut ever, who took up next generation DNA sequencing and gene expression technologies and tested them in flight? And they work just as well, and in some cases better than on the Earth. So I think we're making really good progress in this regard. We have to understand our microbiological surroundings in us, on us, and in the environment. And with that, I'll close my discussions, and I look forward to questions at the end.
Well, thank you very much. Annalisa Paul.
All right, so what I'm going to do today, what I'm going to do is give you a little taste of some of the tools and approaches of using omic technologies and how we can assay some of the aspects of how we adapt to spaceflight environment. And so we use omics research in our lab, and that's one of the examples that I'm going to walk you through. But for the most part, I want to talk about first in the support of exploration, we need to be able to find how we can assay the impact of spaceflight. How do we measure that? How do we tell, what is important and what is not to a biological organism that you can't necessarily always get the great picture of it until you look a little more closely into their gene expression patterns. So one of the things that we like to be able to do is, can we metabolically assay what is the stress points, what are the things that are important for a biological organism to adapt to a long-duration spaceflight that supports our exploration initiative? And how do we address it? And how do we fix those things if we do find them? Well, the major point that we can do is we can use omics tools, and this is the sort of the buzzword now that describes things like transcriptomics, proteomics, epigenomics, that provide a global readout of the genomic responses of an organism to this novel environment. It gives us a window into what is actually happening and what tools are being utilized by organisms as they adjust themselves. There's been a lot of work on molecular biology on the ISS, and yep, 10 minutes is not enough time to do very much in the way of shout-outs. But humans, rodents, invertebrates, microbes, plants, tons of work, and a lot of it is just coming out now into the literature. But another place to look for the things that have been done is to scroll around into the Gene Lab, NASA Gene Lab sites, and you can see what has been uploaded as far as data into this repository. And so I just threw out a few examples, but there's now hundreds in those databases. But I'm going to take a look a little more closely at plants, because plants is what I do, and I've been doing this for about 20 years as either PI or co-PI as a NASA-funded researcher in spaceflight, both in the shuttle and in the ISS eras. The things about plants is that they're really exquisitely sensitive to their environment, and so we're able to assay their molecular responses in a very nice, clean way that enables us to look backwards to their physiology and to their responses to their environments. There have been numerous spaceflight experiments that have done with plants way back into historical era, and in the last 10 years there's been quite a few that have done molecular biology. Variety of species, variety of genotypes. One of the things that we found is that plants all respond very differently, and that their sub-environment that they're in is also very important to the kind of responses that they mount. The experiments that have been all assayed so far, they're all very different. The responses are very different, but each experiment, even though as unique as it is, does have a number of things in common. One of the threads that runs through almost all spaceflight experiments is changes in their cell wall remodeling genes. And so what this means are the genes that function in the stiffness, in the looseness, in the structures of the cell walls of plants. Almost universally in every experiment that's been done thus far, some of the genes are different in the way their patterns they are expressed in spaceflight. not all the same genes, but in the same category of genes. Another class is pathogen response. And a lot of the pathogen response genes are also kind of associated with cell wall remodeling, so it tracks very well. And then another category is reactive oxygen species genes that are associated with the adapting to ROS responses, again, oxidative type stress. So most of the plants that we work with, you have those nice pretty ones in the lower corner of the screen, but really most of the molecular data that we have gathered, we collectively as research groups, are mostly plants grown on Petri plates. This picture in the center here now is Arabidopsis thaliana growing on the surface of a agar plate with the nutrients in the water incorporated into that media. And this is the kind of work that I'm going to tell you about here in the next couple minutes. So there's two stories of spaceflight transcriptomes that I'm going to give you examples. And I want you to keep in mind that although these data are from our own laboratory, these types of data and the types of analyses that I'm going to show you here are examples that can be applicable not only to plants, a lot of other people have been doing plants, but also to any biological organism. These are the tools that we use to inform ourselves of what's going on in space. So if you look at a comparison across tissues within a plant, for instance, and so this particular experiment was the transcriptomes were done on leaves, hypocotyl, which is that little kind of neck area between the roots and the shoots of the plants, and then the roots. And you can see by the patterns of expression in this heat map to the side there, red is genes are up, green is genes are down, that the patterns of even the tissues are very different. So even within the same plant, the gene expression patterns are largely very organ-specific. There's only 17 in the center there that are coordinately expressed among all tissues. So all tissues are using different strategies to adapt to the spaceflight environment. If you did another example of spaceflight transcriptomes, a comparison among genotypes. And so in this case, the same tissue was used in each case, but 3 different cultivars, 3 different genotypes of the same plant. We used a strain called WS of Arabidopsis, a strain called Col0, and then also we took a Col0 strain that had one single gene change, just one, a gene called Phi-D, which functions in some light signaling aspects, and we also looked at the transcriptomes there. And so what you can see again with this heat map is they are very different. They're very different. But the other thing that I want you to pay attention to is that look at the WS column. There's just not many genes that are differentially expressed. And so the metabolic load for going to space is actually quite a bit lower for WS. And what happens if you change a single gene, just one gene, in the Columbia 0 background, and all of a sudden you've reduced your number of genes you need to adapt to spaceflight from 217 to 64 plus 37 there. And this is an amazing thing to us, that we can reduce the metabolic load with just a single gene. So deleting a gene has a huge effect. Changing your genotype has a huge effect. So what can this tell us? How can this inform us? So let's go back to some of the original questions that I brought up in my earlier slides. Is how can we assay? How can we make an assessment of what is important and what is not? So using spaceflight transcriptomes, whether you're a plant or a nematode or a bacterium or anything, gives us that you can look at the differences between the same plant, between the same species, between different organisms, and pinpoint that different genes are engaged to physiologically adapt to that environment. And when you have different genes that are being engaged, that means that different adaptive strategies are being used. So what we can do with this knowledge is we can identify genes that reduce the metabolic cost of spaceflight adaptation, and we can also engineer plants, maybe even nematodes or Drosophila, to relieve the stress to avoid maladaptive strategies. This is, of course, very important when we're going forward in an exploration initiative. So the importance, the implications of plant omic type research for space exploration is kind of threefold if we look at it. First are the fundamental insights that we get, and these are important not only for space exploration, but for exploring our own environments here on Earth. Bioregenerative life force is an easy one. I would say that this is on the critical path, because if you don't have successful bioregenerative life force as we go forward in exploration, you're not going to want to find out that plants are critical to your system when you're already there. The other thing is crew health, both with diet diversity and, for instance, enzyme support, the nutraceutical support, like the materials that Ellison was talking about earlier, and also the outlook that astronauts have that Mary was talking about as well. So all of these things are connected. They're all interconnected. And I'll give you one example in my closing minute. And that is, if we take here and we look at some of bringing the tools together that we've had. So you see the transcriptome map there and all the arrows that I've got going. What we found is that when we look at the transcriptome of some of these genes that were of interest, I've got TOUCH4, SPL1, 5, COW1 down there, all those genes that are being differentially expressed impact how roots grow and develop. And what we found is a phenomenon happens on orbit that's called skewing. And the reason why this is important and why that those genes feed in there like that is because way back in the day, 100 or so years ago, Charles Darwin said, you know, plants need two things to skew. They need gravity, and they need an interaction with the plant surface on which they're growing. So this is a notion that has held since the 1880s. And what we found is that that's actually turned out not to be true. Is plants grow on orbit, they skew on orbit without gravity, and they are able to do this in a way that we never would have found out if we hadn't had a microgravity environment to test those things. And so now, looking forward 100 years, these are the kind of landscapes that we're going to be able to see. But before we can get here, we have to learn some of the critical information that we may not even know we need yet. So plants are critical in the whole life stream of this. All right, thank you very much.
Well, thanks so much for three extraordinary talks that are not only of basic scientific interest, but great help to the committee. I want to start by asking what might be a challenging question of each of you, if each will answer it, and then I'll turn things over to the audience for more intelligent questions. I want to ask, in each case, animal cells, bacterial, microbial cells, plants, what percent of the way are we towards knowing what we need to know to put people on Mars? Who wants to go first? What percent? And I just want your own opinion.
I can start. We're barely at the tip. We're not even at the tip of the iceberg, because going to Mars is going to get into fractional gravity. Nobody knows any idea how life in general, plants, microbes, human, are going to respond to fractional gravity. Right now, all of our data, 99.9% is on the ground, right? Unit gravity. And we've definitely have some data over the course of 50 years in microgravity, but it is puny compared to what we have here. And we show vast differences in how microbes and human cells respond between here and here. Night and day differences, unpredictable differences. So are we happy saying, well, we know all the answers in microgravity, let's just go to fractional gravity? I'm personally not comfortable with that, because I think biology has way too many surprises for us. I think microbes are smarter than we are anyway. And so I think we're not even at the tip of the iceberg for knowing anything about how they respond to deep space.
Putting a number to something like this is interesting, particularly since you're lumping me with animals and humans, but which is an animal. But I think we understand a lot in terms of countermeasures. I think we've got the practical ability to put humans in space for a longer period of time. We know we have issues to deal with, so we have a number that is not zero in terms of humans and in terms of understanding gravity in animals. If I go back to the shuttle era in particular, we understand a lot more. We have a lot. But again, we don't have an integrated program. And so I would say that we're making progress. Tip of the iceberg, maybe we're past the tip of the iceberg, but we have a long ways to go to truly understand what the effects of gravity are in a living system. And I would reflect that back, and I didn't bring it up in my talk, but Earth applications, understanding the role of gravity for us on Earth is still something we don't completely understand, because we do live in a 1G environment. We've evolved in a 1G environment. Fractional gravity is another issue. I think artificial gravity is a program that is now starting to evolve within the agency. There are mutual discussions between SLIPS and Craig. I'm not sure what your new acronym is, but between that and HRP, I think there's an evolving area where they're looking at short arm centrifuges in humans. They're looking at spaceborne centrifuges with animals. Jackson just flew its mouse experiment, its mouse centrifuge. So I mean, we're past a 0 point there as well. But whether we actually are going to truly develop a fractional G environment to live in, going to Mars and back, that's a very open question right now. And so we may be going in microgravity out and back. Again, I think those are discussions that are underway, and so it's not a zero-sum scenario that we don't know where we are. I think we've got pointers to where we need to be looking, but we have a long ways to go.
Well, boy, asking for a percentage, that's really cruel.
Well, I didn't get that from either of the other speakers, though. I'd say we're at 5% and 30%.
Yeah, you know, the
25,
square root
okay.
of pi over 2, I don't know. But as you know, this is we're getting close. We're for sure getting close. Are we a quarter of the way there? The problem is we don't know what we need to know yet, and this is the big unknown. It's like both Chuck and Cheryl said, is that space is indeed a novel environment, so outside that our realm of evolutionary experience, that we know that plants in particular throw a lot of things at microgravity. They pull a lot of things out of their toolbox that are not necessarily useful. And so one of the critical needs that we have in our particular field is trying to dissect away what's important and what's not, and how can we engineer plants to be better supporters of ourselves as we sally forth. And so this is one of the things that I think is really lacking so far, is we're just scratching the surface of now being able to analyze how plants respond to microgravity and beginning to correlate enough data that has come together from various kinds of researchers to start to get a path. And it's going to take a while to fully integrate what all that information really means, so we are prepared to get the nutraceuticals and everything else we need to support exploration.
So 50% maybe.
25.
25, 25, and 5. Okay.
Well, I would say, let me recapitulate something you just said, because I think it's kind of where all three of us are hesitating. We don't know what we don't know.
Yeah.
I mean, and I'll give the example with the astronauts, the VIP syndrome that has recently arisen, and it's become a major biomedical issue with the astronauts. Why are we seeing this shift in ocular and retinal function that is persisting post-flight? We knew that there were some ocular shifts in short-term flights, but it's the exploration class missions that now are bringing this to the forefront. Huge biomedical issue. We had no prior knowledge of this until a few astronauts came back and said, my vision is not back to where it started. And so it was unexpected. It started off with an intracranial pressure issue. I would actually argue that it may not be intracranial pressure based on the work that's coming on. I think we now have, at least in our work with the animals, some issues that indicate that there may be some sex differences, that males, particularly older males, are going to be more sensitive to it than females in general. And so, again, this is what we don't know makes it hard to predict, and which is why the hesitation, I think, for all three of us from the numbers. Who expected the change in virulence? I mean, you tested the change in virulence, but, you know, a priori expectation of, oh yeah, we go up there and these little buggers are going to become more dangerous. You know, there was no expectation that that would be the case. We testing it, we found it out, and now you've got a major research career that, and a number of people are looking at this and trying to understand it. So it's the understanding of what we don't know makes it tough.
Add to my colleagues' comments, which I agree with wholeheartedly, and speaking to some points from the previous speakers that we had. You know, we spoke about fractional gravity, but this is a very multifactorial environment up there, right? Differences in radiation. You have all kinds of different unique environments that the crew is facing up there, from circadian rhythms to the potential for behavioral alterations, confinement. Their microbiome might be different, fractional radiation. So I think it is important that research also look at how the physical and the biological components are, the interrelationship between them as well, not just in isolation.
Okay, those are some great answers. Let's have some questions from the floor, please.
From ADSR Society. about the longevity of the ISM, which is what we're talking about, 15 years or so, 20 years, and arguing could there be evolutionary processes that are growing simultaneously that are having a substantial impact? Do we have any idea of what the trends are?
That's a great question, and we don't have a lot of information on that because there haven't been any long-duration studies for growing microbes up there. Now, we put bacterial spores outside the ISS and let them go for long periods of time and seeing if they can germinate, and a small percentage of them can, but spores aren't growing. And that's why our first study coming up with the long duration, we're shooting for 300 generations in flight, which studies on Earth have shown evolution in terms of lasting evolution, that's more than enough time to have those kinds of processes happen. So we don't. And the health, that's why I stress the health of the ISS is inextricably linked to the health of the crew, right? Because they're interacting, they're interfacing with all the microbes there. The microbes are also interfacing with the control systems. And some of the pictures I showed were damaging the integrity of the material systems themselves. And microbes, I think they've been actually underutilized in flight. They're the most dynamic living systems we have. They have the shortest generation times. These are going to be the organisms that will give you the first readouts of what types of evolution is going on. So that is the direction that more research needs to go. I know that's something the committee challenged last time on the 2011 decadal survey. NASA has started to pick up that baton and pass it on, but we don't know. But I will guarantee you, absolutely they're evolving up there, and they're evolving quickly, and that means we're doing something similar.
Great, thank you. I almost thought I was going to get scooped there. My question is on a topic that none of you really.
By yourself again. I know you asked a question last time.
Sorry, Jeff Smith, ASGSR Society and Kennedy Space Center. None of you touched on this topic. I'm hoping that each of you would be willing to spend a minute on it in your respective field. The decadal survey called for, as one of its recommendations, multi-generational studies. And Cheryl, you just started to answer that. But for plant biology, microbial biology, invertebrate and invertebrate biology, the challenges and complexities of multi-generational studies are very different. And the kinds of important questions that need to be answered that only multi-generational studies can afford are different. And I hope each of you would comment on the challenges and the relative importance of multi-generational studies for your field, as opposed to other areas of research that are ongoing. Thank you.
All right, I'll jump in first. So what you ask is very true for any kind of organism. Luckily, with things like Arabidopsis and bacteria, those generation can be very quick. So an Arabidopsis plant, for instance, can go seed to seed in a couple of months. So feasibility, you could run a multi-generational experiment on the space station. There have been experiments that have gone to seed up on the ISS in both Arabidopsis and wheat. But so here's one of the experiments that is very important from the concept of adaptation, and that is we're just now getting also a handle on epigenetic effects. And so we know that not only do primary mutational effects happen, much like what Cheryl was talking about in, if I'm understanding it correctly, evolving over time and stuff, because bacteria have a very quick half-life. But for plants and for higher organisms, in order to get a heritable change to the next generation, that does take a lot of Darwinian kind of evolution. But we know that plants and also other organisms will respond to environmental stress, sometimes with epigenetic changes. Some of these kinds of changes can be heritable to the next generation. And so one of the studies that have not been done yet, that is actually quite critical to be done, is to look at how a generation of, say, plants, Arabidopsis plants, grown on the space station, what kind of epigenetic changes do they accumulate, and then take those seeds and send them back again compared to the controls and see what kind of adaptive metabolisms may have been adopted.
I think the issue with animals, we're not ready for humans yet, but I think the issue was raised. I know Ames, Jeff, as you're aware, was looking at developing a multi-generation capability with the rodent research facility There's a plan in place to start looking at that kind of an issue, and again, it becomes an issue of priority and funding. The word was mentioned quite a bit so far this morning. The time constants are different because they are longer generational, even with the rodents. And they are going to be important because not only are we looking at issues of heritability, but in cases of mammalian studies, we're looking at issues of reproductive integrity, both in terms of the males and females. We're looking at impregnation, we're looking at conception, we're looking at gestational effects. We touched upon this a little bit in terms of gestation in the shuttle era with NIH R1 and R2. But again, it's a program that would be important. We need to understand it, particularly if we're going to be sending men and women in space in the long run. We do have indications that heritability of G sensitivity, if you will, I'll say that very generically, is present in mammalian studies. Both Jiro Oyama and Milt Smith previously, our prior generation, with ground centrifuge work showed heritability issues with multi-generations of animal study chronically at plus G. But again, these are areas where there have been long-term plans that have been laid in place, but there's no execution activity at this point in time on them.
From the perspective of understanding infections or even microbiome changes, problems with using human cells long-term are that, to my knowledge, and please correct me if I'm wrong because you're more integrated into this than I am, I don't think there's any hardware that's been developed that will support long-duration cultivation of human cells under the appropriate environmental conditions and volumes needed. I don't think it exists. You can also, why don't you can use human surrogates. You can use C. elegans, you can use Drosophila, you can use a number of things. That's true, you can. But the organisms have to be capable of being stored long duration, because it's not just when you go up to ISS now, they can be unpacked. And for example, our experiment, we needed those C. elegans nematodes to be unpacked and used because we had a 15-day timeline, and after that, the viability went down the toilet. Okay, and we needed to make sure everything was in the same phase of growth, because you cannot waste that precious resource of spaceflight, ever. Can you justify that? And we don't want to be infecting hosts that are in different phases of growth. So what difference does it mean when we do it on the ground, when it could just be simply a phase of growth that the host was in and not at all related to the actual environment itself? So those all speak to, these are things that can be worked out. So my glass is really half full, even though it isn't, it is. These are things that can all be worked out, but those are the kind of critical thinking problems you have to think about when you're having your engineers work with the biologists. Don't develop the hardware first and then ask us what the heck can we do with the box later. But they need to synergistically work with biologists side by side. We do that, and we have helped them develop 3 new sets of hardware that, through those, as you well know, because you were there, you were a big funder for Micro 5, that can now be useful for these kinds of studies. It can happen, but again, that requires consistent and stream of funding, an appropriate stream of funding to make that happen. But you can do it, Jeff. You can solve everything. It can happen.
I'll have to ask Craig to do that for me now.
So I had a question. My name is Jamie Foster from the University of Florida and ASGSR. I wanted to add another layer of complexity with how we overcome our ignorance about what we don't know and so forth, and that's microbial dark matter. And by that I mean, we're generating lots and lots of big data sets, and we only know a fraction of what these genes do and what they are, and also we only know a fraction of what the microbes are. So and I don't know what salmonella is, but in E. coli, we only know that's the best studied lab rat microbe out there, and we only know about 70% of what the genes do. So we have to start to dig into the data we do have and really look for patterns of the unknowns and start to elucidate what some of these functions, because I know in my own transcriptomic data, the most differentially regulated are always unknowns, right? And going after them is hard. And so that's something that we're going to have to think about as a community, to tap into that unknown material.
Jamie, first of all, it's good to see you in person. I've wanted to meet you for a long time. I've been following your work. It's beautiful. I want to add on to your comment because it's very prescient and it's critically important. Not just what you said as well, but we have got this beautiful database of omics that NASA has spent a lot of time and effort and beautifully made available to the public, to scientists, that we can scour and we can look through things. And that is fantastic, and we do have a tremendous amount of it. There is a problem when it comes in doing that to context and standardization. So you know as well as I do that and by the way, Salmonella is in the same boat as E. coli. It's the best characterized pathogen, and we know a lot, but we don't know a lot about it. So I can go out there, and more groups are doing this. They're scouring it, they're developing new algorithms, new data processing software systems to try and come up with unifying themes, okay? And that is great, that should be done, but the problem is context. You have to be very careful when you're drawing conclusions about that, because very different types of media, very different types of growth phases, very different types of temperatures, very different types of when you're how you're fixing those samples and taking them and processing them downstream. And it's not just spaceflight omics that faces this, all omics face this. This is why we have millions and millions of biosignatures for cancer and any other disease you can think of sitting in databases, and how many of those have actually gotten us to a translatable product? Not many. All right, so context matters, and you can never grow cells, any kind of cells, under all the same conditions and all the same parameters. But I really strongly advocate for a program, especially within NASA, where you could have maybe a set of experiments that are piggybacked off other experiments that multiple investigators can do together. We've done this before. You and I could easily do this with the kinds of organisms we use, where all the conditions are standardized. So number one, it saves NASA resources because they can use the same hardware, they can fly together, they can piggyback using their experiments of interest as long as they fit within a certain parameter of environmental conditions. And then you can have the same snapshots in time. You can have everything be the same. It's not going to solve everything. But I do think that's critically important. And in terms of the microbial dark matter, that everything turns up, or generally the exciting things turn out not known, I think we can expect more of that, because for Salmonella, we had a significantly hotter bug, significantly hotter, all right? And not one known virulence factor was upregulated. They were all downregulated, okay? But we had a lot of genes that we don't know their functions, which heretofore had never been shown to be involved with the pathogen, or causing, with infectious disease. So I think your question is so big and so important, it can't be solved here, but it's a very important point to bring up.
Do we have time? Yeah. Okay, so on the other side of the coin, I suppose that there is dark matter in Arabidopsis as well. A good portion of the Arabidopsis genome is also encodes genes of unknown function. But one of the things that, for instance, that we, in our experience, that in a normal stress kind of response that you might find on the ground, so say we call salt stress in Arabidopsis, you might have something in the neighborhood of 25 to 30% of the genes that get transcribed into response of that be unknown, quote, unknown characterization. In response to spaceflight, about 42% of all the genes that were differentially expressed were unknown. And so part of this, we think, is because of this, again, this whole novel environment business, is that plants are just throwing all manner of tools at this environment, and they're not quite sure what they're pulling out of their toolbox. But 42% were unknown. So one of our graduate students took a whack at one of these genes that were very highly differentially expressed, and to make a long story short, she has defined the function of a new gene that heretofore was unknown. It turns out it functions in oxidative stress signaling that was important in spaceflight, not important on the Earth. We never would have probably come across what this thing did if we hadn't sent these guys to space. A lot of genes are probably like that in this dark matter mass, that we won't know what they are until they're revealed.
I'm going to make one comment as a not related to omics per se. I only dabble in that field, but this comes when we talk about animals, and particularly with humans and crew, into a question that Elliot, you originally asked me in terms of monitoring. And I think one of the things that's important is to establish an understanding, as you said, baseline standardization. But when you have, let's say, 10 mice in a cage that are group housed, and you have no idea about biological variability or individual responses, and lumping all of those animals together as a generic response is clearly not a good idea. And I think instrumentation to be able to monitor individual subjects when they are large enough is appropriate. And I think this actually reflects back to where I was inferring for the biomarker question earlier in the last panel, because while yes, it's too late to do biomarkers when you think there may be a problem, you need to do it earlier and understand the shielding or whatever. But again, within a crew, you are going to have biological diversity in terms of sensitivity to response. And to be able to track individuals during a mission at some level, with some level of standardization, I think becomes important, because it is preventative for those individuals, but it also allows you to expand your database to understand which individuals may or may not be sensitive and may have to have additional attention to detail. So that's at the applied level. But at the research level, even with animals, we still need to have the ability to collect individual information about each subject, so that when you do omics or any other kind of averaging technique of any kind of biological measurement, you have an idea of what the response was across the population of your study.
I think we're going to have to stop at this point, because we've used up our time. Additional questions we can take at the next break. I want to thank the speakers for tremendously interesting and informative talks, and then turn it over to the next session.
While the next panel is making its way up to the front, I want to take the co-chair's prerogative to pull on something that Chuck was mentioning just now. And that is, one of the things that is being exposed by doing these number of genotyping and genomics associated experiments in space is that individual genes and individuals will be reacting differently to spaceflight.
Hello everybody, I'm Reza Bastian from University of California, Riverside. It is my pleasure to introduce our next speakers, which we are going to change the gears from life sciences to materials and combustion. I have three distinguished speakers. To my right is Professor Martin Glixman, who is presently Dean of the Engineering at Florida Institute of Technology. He also holds the chair of Allen Henry at that university. Marty has had 3 successful space experiments in 1994, 96, and 97. He's going to talk about his work somewhat, but his research has set some of the most accurate data in materials processing that I will let him go over that briefly. Next to Marty is Professor Carlos Fernandez-Pero of the University of California, Berkeley. He also holds endowed professorship of Almi Maynard and Agnes Ofield Maynard endowed chair in mechanical engineering. Carlos also serves as an associate dean at the graduate division at UC Berkeley. His research is in heat transfer, mass transfer, combustion, microgravity combustion, and ignition and flame propagation. Of course, he has numerous publications and research projects that are related in this area. Last but not least, speaker is Frank Urban, who is branch chief of combustion and reactant systems of NASA Glenn. In fact, anytime you want to know know about what will happen, you call Frank. So with that brief introduction, oh, Davis, sorry. You call David, not Frank. The questions that they are going to be addressing is basically what we know in materials and combustion areas, and also what we do not know. Are the research that are going on now going to be enough to give us confidence? Confidence meaning in engineering terms over 99%. Okay? If you go Six Sigma, even beyond that we will have a successful mission and all the goals that are set forward. With that brief introduction, I would like to ask Professor Marty Glicksman to start the presentations.
Thank you, Reza. Thank you. I just want to recall a remark that Gail Allen made this morning about push and pull research. And I think in very large measure, the work we did back in the shuttle era, a good bit of it was push research, and quite a bit of it, including my own, was kind of this pull research, which really was directed to a slide she put up on physical research organizing questions. And I pointed out in my question to her, how do structure and complexity arise in nature? And that's a very, very broad problem, cuts across biology, physics, and materials. And just to remind the audience too, Charlie Linoli talked about dendritic spline damage in charged particle irradiation affecting cognitive degradation, where he showed how the pattern of these dendritic spines would be affected, and indeed seems to have some important results. So I think what I'm going to try to relate back to is ultimately something very practical, and I'll just throw this out in the beginning, though I won't drill into it, and that is habitation. Habitation on these future missions will require the use of in situ materials. You cannot possibly contemplate bringing the materials up to a place like Mars or even the Moon, where you're going to use that in situ material for protection and habitation. And that requires processing this material, turning it into something useful. So, on my talk, I want to talk about the fact that we've always loved interesting patterns, and the hand sketch on the left I picked up when I was visiting Germany about a year ago at the Bundesanstalt für Materialforschung in Berlin, and there was this beautiful hand sketch done. You probably can't see the fellow's name, but let's see, how do you turn this laser on? Yeah. Oops, and I have to go back again. Sorry about that. It says right here, Adolf Martens. And he's the guy who, he's probably the most famous steel metallurgist and has a famous phase called Martensite, eponymous phase named after him. And metallurgists, as well as biologists and all kinds of scientists, love patterns. And you can see on the right a snowflake, which shows the sort of iconic pattern of winter, and what drives that. And it turns out there is a relationship between what forms that dendritic crystal on the left hand sketch by Adolf Martens back around 1885, what forms that particular pattern at these small scales, we call mesoscales, because they're not really microscopic, they're not quite macroscopic, and features of a snowflake from the Smithsonian collection. And the standard paradigm, which has been around for about 40 years, is, and I'll sort of point this out as we go on to, I'll have to show, if I can, a video. I want to show how it cuts across so many different fields. Here is a picture on the upper part of the blue morpho, and that's a beautiful tropical butterfly, and nature has apparently given this butterfly a remarkable nanostructure which is based on these photonic dendrites, which in the electron microscope show up from the scales of this butterfly's wings. If you look at a blue morpho, it's not blue. It's a rather dull brown. And the blue comes from this nanostructure, which is built into the wing. And it turns out white light comes in, blue light goes out with about 95% efficiency, and you get this magnificent blue butterfly that doesn't have a speck of blue in its wing. And people are obviously interested in it for things, as I've indicated on the slide, to make chemical probes, fast thermal detectors, and people even foresee pigmentless paints and coatings that will never fade, because this is a photonic effect, not something that will be affected by fading. On the lower part of the slide brings you back to materials, and lo and behold, again, I show 3 superalloy turbine blades from about a 40-year period of development from polycrystals, the one on the left, directional ones that were developed in the late 1960s, and finally single crystal monocrystals. And let me tell you the difference. The one on the left, that polycrystalline blade, was flown in early military jets, and the pilot got about 30 to 40 hours on his engine before the engine had to be torn down. The monocrystal on the right is what you fly on in a commercial jet today, where you get about 25,000 hours of operation of the turbine, and that allows your ticket to be as cheap as it is, and the efficiency and so on. All of that coming about by a pattern change developed through these, in this case, this dendritic forest, which was exposed when one of these blades, which is solidified, the mold broke, and a sharp guy at Oak Ridge by the name of Stan David took an electron micrograph of what is growing to make that monocrystal, and it's that dendritic forest. So what produces that? And as I mentioned, for about the past 40 years, the physicists have told everyone it's noise, that there is no information stored in the molten phase. How the butterfly does it, I don't know. But in the case of these metallic patterns, there is no information at a level that can produce those kinds of patterns. So is it really, the physicist said, well, it's got to be noise because there's nothing else. We performed an experiment, 3 experiments actually, back in the mid-1990s on the shuttle. And I wanted to compare how we operated the shuttle microgravity payloads and a few experiments versus the ISS laboratory, and how things have sort of changed and progressed. We had ground-based scientists, and notice they got my hairstyle correct as a ground-based scientist, and we would control everything from our laboratory. I had a team of students actually doing this. We call those payload experiments, and NASA was willing to invest literally years of development for custom apparatus. My equipment weighed about 760 pounds and was a primary payload on the shuttle, so we got to write the flight rules and so on, as opposed to what I call a laboratory experiment, where you trained an astronaut with a generic piece of equipment to carry out the experiment on board. So in the payload experiment, you ran it from the ground, and we were very fortunate. We really had 3 experiments and 3 home runs. Now, I'm not going to go into the complexities of communicating with your experiment, but the round-trip signal, which is indicated by that complicated diagram showing we were up at RPI up in New York State, and all the ups and downs to actually reach our experiment. But I just want to point out that the latency period for that, controlling the experiment, where we would put in a temperature change or what have you, was about 1 second. Compare that to changing something going on in Mars, where it's about 25 minutes. Completely different feedback and operation. What I wanted to show you, though, was a K-band data stream that we got, the very valuable data coming back. And is it possible to play that first video? Yeah, thank you. And coming out of this little capillary tip in the middle of the program. Is there a problem? Oh, we had it embedded. Okay, we'll have to skip it. It would show you a nice dendritic pattern forming, and that pattern then could be analyzed. We would receive the data stream down. This came from STS-87 at the time, and you get this beautiful complex pattern. What we used the pattern for was to check a thing called the scaling factor, which selects, based on noise theory that we were testing, what does N mean? Am I done? Okay. Well, wow. Okay. What the data stream gave us was essentially the fact that this theory was incorrect. What I'm going to say just in conclusion, really, from my talk, is this pattern, which came out of our third experiment, showed us very clearly that the noise concept was wrong. What's developed over the last 20 years, and we're getting ready to really push this ahead, is that we have discovered a thermodynamic field that we do believe causes these patterns, gives us a whole new paradigm for understanding pattern formation, and is an extension of this pull-type research that NASA was willing to fund these many, many years ago. And it's telling us, going back to the habitat issue I brought up, that to produce useful materials in a partial gravity like Mars or on the Moon, we have to understand the pattern formation as we sinter them into useful materials or otherwise process them. And the payoff in this particular level of NASA research from Glenn has taken about 20 years to really yield some dramatically new information. Thank you.
Next, we have Carlos.
Yes, so I'm going to talk about combustion in a spacecraft environment. But I mean, combustion is a very varied field encompassing from power generation to materials generation. But today I'm going to talk primarily on an issue that we believe is critical in a long-term space exploration, and that's fire. So the potential for a fire in a spacecraft is serious, but what is more important is that the consequences can be catastrophic, not only from the point of view of the heat generation and the damage from heat, but also post-fire, the effect that the soot and the corrosion can cause on electronic systems and different operating systems in a spacecraft. So we call that really the first aspect of the fire is the potential generation, and in that regard we address that by looking at what we call the flammability of combustible materials. And flammability, we address the issues from potential ignition of the material to the spread of the fire of the material, the heat generated during that combustion process, and the potential subsequent effect on adjacent materials. And the problem is that from the point of view of the safety of space travel, is that in future spacecraft, the environment is going to be very different from that we have in normal gravity. So we understand quite well, the quite is questionable, but anyway, we're fairly well the behavior of fire on Earth. And the problem that we have is that in a future spacecraft, not only we are going to have a microgravity environment, but the HVAC, the air conditioning system of the spacecraft generates very low velocity flows that they are not achievable in normal gravity, because buoyancy, we are talking about a combustion process when you have heat generated by the combustion process. Those high temperatures together with gravity induces buoyant flows that changes the characteristic of the combustion process. So that is going to change in spacecraft environments because of the low velocity flows generated by the HVAC system of the spacecraft. In addition, NASA is planning to have elevated oxygen concentration, and as a consequence, or the opposite, low pressures in the spacecraft, and as a consequence, higher oxygen concentrations to keep the partial pressure of oxygen at Earth conditions, so that for the comfort of and the life of the astronauts. That's called a normoxic environment. So here we are facing a problem that is the onset of fire in an environment that we have microgravity, we have low velocity flows, we have elevated oxygen concentration, we have reduced pressure. And those physical processes are affecting the basic mechanisms that control fire or combustion. It affects the heat transfer aspects of the problem. It affects the thermodynamics of the problem. It affects the fluid mechanics, and it affects the chemical kinetics of the problem. So here we are dealing with a number of circumstances of which we have very little information to date. So now I was trying to justify the potential scenarios of the fact that a fire is a possibility in a spacecraft. And I remember years ago when I started working on this problem, one of the first big conferences that we had on combustion in a spacecraft, a professor from Penn State years ago, Faith, he stated that the probability in a long-term space travel of having a fire is 1. Right? And so anyway, these are what I'm talking. This is the problem that we have. We have a number of environment variables in a spacecraft, and we have to affect. So how do we quantify material fire safety? So we have ignition. We have, as I mentioned, flame spread. Flame spread is the propagation of the fire through the combustible material. The burning rate is important because that gives us, as a result, the rate of heat release that is affecting how the fire propagates to different objects. And in addition, one thing that is missing here that I have left it for David Urban to address is that the aspect of the fire, that heat release is one of the aspects of the fire, but in addition we have the products of combustion, and those products of combustion often are toxic, they are corrosive, and they also, it turns out that they are conductive, so that when we have the position, for example, of soot on electronic components, we have short circuits in the electronic components. So I think that I was going to talk a bit about how much we know. This is a question that has been brought here, is how much we know about fire in a spacecraft or combustion in a spacecraft. And we are starting to be at the tip of the iceberg, trying to understand some of the problems. For example, here there is a photograph there on a flame. This is a polymer burning. On the left is in microgravity, in normal gravity. So you have the buoyancy lifts the flame, and we have actually a reddish color flame. It's a fairly high temperature radiating flame. And on the right we have a microgravity, the same material burning in microgravity. And we have first, you can see the difference in shape. That difference in shape affects the fluid mechanics, the heat transfer, and the transport processes of the combustion or the burning mechanism that cause the burning of the fuel. Also, the production of products is different. That's why we have different colors of the flame in addition of the temperature. So anyway, there are a number of mechanisms that we will not know, and of course the final question is, do the materials, combustible materials, have a higher probability of a fire in a spacecraft environment or not? And what we know right now is that there are some aspects of the problem that they are enhanced. For example, the time is easier to ignite materials in a spacecraft environment. On the other hand, it seems that the rate of flame spread is reduced. And also the limiting conditions for a combustible material to be flammable are extended in a spacecraft environment. And I have the same sign, the end, so I meant it here. Thank you.
Next is David Irvin, just to make sure that I get that right this time. He's the person that you're going to call. I don't know how he's going to get there. Yeah.
There we go, Frank.
David, please.
Well, thank you. I'm, as he said, I'm the discipline scientist for combustion for NASA, and also I'm focused on both applied combustion for terrestrial applications and also for spacecraft fire safety. And this just shows the wonderful logos. We spend probably a little too much effort on our artwork, but for the ISS series of experiments. And combustion is important in low gravity for study, both for the terrestrial applications and also the spacecraft fire safety, which Carlos discussed. And you sort of wonder, why does combustion matter so much? Every flame you've seen knows which way is up, and so there's a gravitational component that's very obvious to you. And combustion is wildly complicated. It involves fluid flow, radiation, fluid flow both in gas and liquid phases, radiation, heat and mass transfer, condensed phase and gas phase chemistry, which can go into the thousands of reactions, and on phase change. So there's a lot going on, and peeling that apart is hard enough, but it's made simpler by the low gravity laboratory in many cases, because it simplifies the fluid flow enough to let us get at some of the chemistry issues we really want to address. This is what we did in the greater than 5 years ago. I just want to pass over that we've been at it since early on the space station, starting with some spacecraft fire safety work with the smoke detection and looking at simple gas jet flames But most of our work has been in droplet combustion because largely that was the facility we were most ready to fly. And the funny story is, this is an experiment that started really as push and really is now becoming pull. And so there you can cross the Rubicon there. And in the beginning, it was the one we could fly. We flew it, and we'd already flown in the shuttle. I wasn't sure how much exciting stuff we'd find. And as you'll hear, it really was quite a bit. So in the most recently, we've continued with that because our next apprentices weren't quite ready. But we've also now gotten very excited about more excited than we would normally have been about these droplets, as I'll explain in the. So the droplet combustion, and particularly what we call cool flames, but a series of other collaborations, including collaborations both with the Italian Space Agency and the Japanese Space Agency, and Flex ICE-GA is Italian Green Aviation, and the 2Js with the Japanese JAXA, also working with them on their facility, the group combustion experiment. This gaseous experiments I mentioned previously were in an old shuttle era piece of hardware, which is what we had that we could fly when asked, and we were able to then convert it to do solid fuel combustion experiments, which have been tremendously successful and gave us a lot of new information on material flammability. And I'll touch on that, which is Carlos and others have been worked on, and Professor Tien. And finally, most recently, we have broken in outside the space station envelope into the cargo vehicle supporting the space station with Sapphire, or a spacecraft fire safety demonstration project. So, and future plans, we hope to shortly be converting over this summer to look at gaseous non-premixed flames back in our combustion integrated rack. So 100-liter chamber, looking at critical issues in flame structure. And this supports primarily terrestrial energy efficiency and pollution. It's on the push side. But we don't know what we'll discover. We have some things we would expect to find, but we were very surprised by the MDCA facility when we spent enough time in there looking at droplets. So I expect to be excited again in the future. And then moving on after that to solid fuel flammability using another insert for the SIR called SOFI, and hopefully to get we're beginning the buildup of a new facility for the MSG because we had so much success with a simple facility made for the shuttle. Why not make one for the MSG? So it's called the Windy with the mu. You flip it around for gravity. Very cute. And then finally continuing on with Sapphire. So why do we care about combustion and low gravity? And I think the line I like to say to the rest of the world is, in engines, just like stand-up comedy, timing is everything. If you think about an engine, your piston or your jet engine or your rocket engine, the fuel's all got to get into the combustor and then ignite and then get out of there in milliseconds. And so that means you've got to get your fluid flow, your mixing, your compression, and your ignition to occur when you want it to occur. And it turns out that doesn't always work out, partly because the fuel isn't always what you expect. And so in your car engine, when it doesn't work out, it's called knock, because that's early ignition. And if it's a diesel engine, you need that early ignition or the cool low temperature chemistry. And so it turned out our microgravity cool flames experiment, as I'll describe, provided a novel means to test this low temperature kinetics in an actual flame system. When it first happened, no one believed it was, we weren't looking for it, and these results were initially highly controversial. The investigator team was highly divided about whether this was really occurring, partly because we couldn't see them. We weren't looking for them. And so, but we saw very strong evidence that something was going on that we couldn't see, and we couldn't see the flame. So this, after much effort, they convinced some of the team, and then they went out to review, and some reviewers initially resisted strongly, now are also very strongly behind, and now there's a whole bunch of other investigators looking at a novel way to stabilize these cool flames. We didn't discover them. Humphrey Davy discovered them over about 200 years ago, but in the past they were always seen on the ignition phase of a flame when you're going from hot gases to suddenly it bursts into flame, and there's that low temperature chemistry in the front phase. We were seeing them in the extinction phase, and we could stabilize them for very, very long periods of time, which had not been previously done, and gave you a real opportunity to study them. And this shows most recently, because of that excitement, we flew, we modified our SIR in the last minute and flew a new camera, and that lower right video is, just turned into the cool flame. It switches detects the extinction of the hot flame. So it starts out with a hot flame, which is the blue flame you're used to, and that's the top three images are a hot flame, that's the flame, visible radiation, this is the droplet, backlit, and this is the image of the hot flame. Then with that same camera, you see no cool flame, so there's no picture. This is the droplet, and this is the image of the cool flame itself. The bottom box is the same camera's using these two things, so we flip the filter. When we see the flame go out, we flip a new filter in place, so we now see the formaldehyde emission, that's the igniter's retracting, and then that's the hot flame, and it very quickly goes out. And that's quite bright, even though it doesn't seem that way. And then suddenly we have to highly switch also integration and image average to get that cool flame video. That's the first time we've seen this, actually seen the flame, and continue with that kind of resolution. So now we can start studying what's going on in that flame. So it's been a huge success and very, very exciting. Otherwise, in solid fuels, in this MSG facility, we use this BASS facility, and we have a huge amount of success for this facility. We've got 55 publications to date. We demonstrated what we believed to be the case. We hadn't really been able to trace it, that there is enhanced flammability in low gravity of materials. And so this particular material burns at 16% oxygen in 1G, and you can't go below that because the buoyancy causes this flow. If in low G, you can go below that amount, and you can go burn it 2.4% lower. So you go 16 to 13.6. And so that is significantly more flammable than it would be in 1G, and we've demonstrated that for a total of 3 materials in space. So these are all relatively flammable materials, but it suggests the other non-flammable materials that we hope to have are somewhat more flammable than we believe based on 1G testing. So this is strongly suggests we need to continue studying it. Beyond that, fire is beyond the complexity in the flame itself. When it's interacting with a vehicle, it becomes vastly more complicated. And so we really need to understand is the threat to an astronaut in a spacecraft vehicle. Every building, plane, train, automobile, submarines, ships, we've done full-scale fire testing here on Earth. In space, we haven't had that opportunity, so we don't have the understanding of the interaction with the vehicle. So this started a series of experiments called Sapphire, using the Cygnus vehicle. It goes up as inert cargo. After it separates, full of trash, separates from the space station, we ignite a flame. Now it's substantially bigger than before. We're able to study it. It takes days to download it because it's a cargo truck, not a research platform. And the first 3, the last one just launched, will operate in July. We're just looking at material flammability because we had to really learn to work on this platform. Going forward, we're going to do 3 more and look not only flammability, but look at the fire detection in the vehicle, post-fire cleanup, and have a greater impact. We're going to let us release more energy into the vehicle itself, so we can really look at how the vehicle responds to that impact. And this just shows you we're surprised even nevertheless. This is the flame now. The previous flames that we did in the glove box were this big, down in the corner here. Now we've got it's half a meter by a meter. This is the flame spreading. Surprisingly, it spread when the wind is going up. It was substantial. This is our case there, and this is a second flight. Substantially slower than the smaller samples we've seen in the glove box. Turns out it's really attributed to the larger flow duct. It allows the flame to be further from the fuel, and therefore less heat transfer to the fuel is what our supposition is. And the big problem was that while a realistic fire, we didn't let energy out of our containment to get to the vehicle. So, key research areas based on input from the workshop that we had in 2014 that brought a lot of combustion topics and fire safety stakeholders. The big thing came out of the push from the droplets is that need to go to high pressure combustion to look at the same kind of chemistry issues for the future engines, all 3, rockets, jets and diesels, all want to go to 100 atmosphere to get higher thermodynamic efficiency, and look at the other vaporization and spray behavior that occurs in these conditions, and the low temperature chemistry. We're building a huge collaboration with the Air Force, the Army and DOE and the industry to build an experiment. We hope to get the space station before it's done. If not, we'll have to find another carrier. And the spacecraft fire safety remains a strong area of need, and particularly getting into fire growth detection and finally suppression. And we need supported ground-based testing and modeling to carry this forward, because you can only do so many tests up in space. Thank you.
Before we open up for questions from the audience, I had a question from the panel. Can modeling or other computational techniques be used to predict behavior or properties?
Well, from the standpoint of material structures and material interactions with the environment, there's been tremendous progress over about the last 15 years in materials, to the point where we have methodologies, things like phase field level sets and other types of numerical procedures that are sort of consistently based on thermodynamics and will really be extremely predictive in how things behave. And even in the area I was discussing on pattern formation, we get very good coincidence now between what these programs predict and the thermodynamic fields that came out of the NASA studies that I mentioned. So it's very encouraging. A lot of sophistication, I think, is available, and I think it's getting better literally year by year, and the techniques and the types of predictions for lifetime performance properties, the relationship of properties to processing, all of that is getting very predictable through advanced numerical techniques.
Yeah. Now, in combustion, the problem from the point of view of modeling combustion is the complexity of the problem. So we have, under actual circumstances, we may have a turbulent flow with a chemistry that incorporates hundreds of chemical reactions, and the computer capabilities that they are needed for modeling are unreachable at this point. And so we are approaching the problem often addressing maybe the thermophysical aspect of the problem, the heat transfer and the fluid mechanics. And I don't know if it is possible to have my talk. There is a slide. Could I have my talk and I can just show briefly a result from modeling? The problem generally is that we either can address one of the aspects, as I say, the thermophysical aspect of the problem, or the chemical aspects of it. So this is just an example of work we can do with modeling. So here is it's just the propagation of a flame over a solid combustible. On top you have, in normal gravity, you can see the buoyancy lifting the flame, and in the bottom you just turn off the gravity, and then you have the rounded flame that you had in the. So we are capable of modeling fairly well the thermophysical aspects of the problem. The problem here is that the chemistry is a very simple chemistry that includes in this there are rather, we cannot use 100 reaction or whatever, or hundreds or several hundreds of. So here, this one actually uses one what we call a global reaction. There is one step further that it can include 4 or 5 chemical reactions. But the main problem, I think, is computational capabilities. Plus, in addition, we don't understand very well the chemistry. What David was talking about, the cool flames. One aspect that we have here, when we have very low velocity flows and diffusion starts controlling the transport mechanism, then the chemistry of the reaction may change. So we don't know, we don't have much information about that. So the answer is
David?
Yeah, so I guess, well, the big answer is if you really thought the model was perfect, good enough, you wouldn't fly the experiment. But the answer is, in many cases, it's an adjunct. Shortly before that first flight of Sapphire, when it spread really slowly, one of the collaborators said, by the way, I think your timing may be wrong. It's probably going to spread slower than you think. And they were right. But on the flip side, that's Professor Tien's team, actually. The cool flames, nobody in the combustion industry world predicted we'd see that phenomena. And suddenly, because the chemistry that we had didn't suggest it, and suddenly we got it, and then they went and tweaked the kinetic parameters, reran it, you could then see it. So it's really, it's a great tool that interlocks with the research. We help refine, in our case, we're mostly refining our ability to model. Not replacing modeling can't replace it today.
Thank you. Okay, we open up now for questions, please.
Thank you. Erica Wagner, Blue Origin and ASGSR board. David, this is really a programmatic question for you. In the 2009-2012 timeframe when the original decadal was written, we didn't predict cool flames and we didn't foresee the availability of missions of opportunity like you've had with Sapphire. What is it that the mid-decadal can do that gives you flexibility to take advantage of those things as they pop up over the next, 5 years?
Well, one thing is to encourage the base research to keep, so we have a research underpinning, so we're ready to respond and find new things that we should be addressing. I think the problem is through the 2004 to till now, I've really, a lot of the experiments have been based on stuff we had already in the queue we've been talking about in 2000. And they said, well, what can we fly now? Well, we've got these few experiments left. We couldn't fly them before, we could fly them now. They really haven't had a very strong opportunity to get new, strong ideas other than the cool flames which sprang out of the program itself. So that's the first thing, is to encourage NASA to have a strong research underpinning to feed the program rather than live off what it's got and the old ideas that I hate to say it. And that's the one. And have to be responsive, you have to have a robust budget.
So this question is to Dr. Fernando Peo. I am a Swami. I'm a professor at the University of Pennsylvania, member on the governing board of ASGSR. So you had mentioned that there is this, in the computational modeling, the difficulties are with significant number of reactions happening. But recently, this computational singular perturbation methods introduced by Harvey Lamb, and then followed by Goosis and others, in conjunction with advanced numerical techniques, probably would be very beneficial in analyzing microgravity combustion. And I would suggest that funding in that area of merging the sophisticated methods of analytical methods of getting reduced set of reactions together with the prevailing advanced computational methodologies will go a long way. What do you think of that?
I really don't have an answer. It sounds good to me, but I think that one of the always I had this feeling that combustion goes behind other areas. For example, in turbulent combustion. So we need the progress in fluids, in turbulence modeling in fluids, in order to proceed. If you add combustion, it's a very non-linear process. The heat generation, the consumption of a species and the generation of heat is very non-linear, and that complicates the job. So you add one more very serious complications to the. And finally, one of the problems, and that's what I would think of is a problem of your comment, is that Often we use experimental data to determine the chemistry, the chemical parameters that control the process. And for example, in low temperature chemistry or very low velocity where transport is reduced significantly, we don't understand very well that chemistry. So I don't know if I have answered your question, but
Thank you.
Generally, the field is split in terms of modeling between the physical aspects, thermophysical aspects of the problem and the chemistry of the problem. And joining them together is difficult.
Thank you. Great panel. My name is Cindy Martin Brennan. I'm the Executive Director of ASGSR. If you don't know the acronym, it's the American Society for Gravitational Space Research. In particular, this question is for David. When you were talking about kind of your future experiments, you had made a comment saying if we were able to fly. Can you elaborate on what that, if we are able to fly, what that is, what that means to you?
Well, that's that particular project is one we're developing, and we're trying to get it in the funded stream for starters, and then we have to meet the deadline for the end of the station or find an alternate carrier. So basically, this is the new science for us. It came out of that 2014 workshop. It's an area where we did not have an underpinning of work in the drop towers because that's pretty much stopped. And so we have to develop the capability to do this fairly hard experiment in a short period of time with limited funding, and yet we have a strong interest from the outside, which we're trying to use that to leverage our funding problems. But we've got to bring it together, get an experiment defined, get it built, and get it up there in time before the planned ending of the space station, and we got to get funded first.
No?
Rob Furrow from the committee. Again, a question for David. The innovative use of alternate carriers or alternate platforms is something that is something we've always considered, all of us have, but is now becoming really interesting in the potential post-ISS era. So you tapped on that. I want to get back to process-wise, how did it come to be that you all, whoever you all is, figured out that you could use the Cygnus, a dead vehicle, to do experiments? That, you know, you pass that off as just, yeah, well, we did this, but that has to have been out of the box somewhere at some point in time. Can you take just a minute and tell us how you got to the point where you do that?
I can actually take credit for this, but I had the first inkling of the idea came to me. I was at a thing at Marshall Space Flight Center hearing it talk about the genesis of the Skylab program. And that was actually some engineer at Marshall's idea. And the original concept was to launch the Skylab full of propellant. It was going to use that was going to be the propellant to get it into space. And then they vent it to space and backfill it with air. And if you look at it in the Smithsonian, that's why there's a big grating in the floor. That was to let the propellant go out as it fired its way up there. And then we canceled the Apollo program, had leftover Saturn V, so we could boost it dry, so you could put more stuff in it, so it was more convenient. So, but suddenly the use of these large volumes lit off an idea in me, because I'd been wanting to do large-scale fires at some point. So then I started looking around for vehicles. I looked at the Progress module, and that just looked too small and crowded. It's the Russian disposable spacecraft. So tried the new Ares upper stages, and I got told to go away because they didn't want any FOD in their tanks. And so, started looking around for other spacecraft. We talked to the Europeans and the ATV. It didn't work out on the schedule. But then combination, having presented it a number of times with Gary Ruff, my partner in crime, to various groups, there came around an opportunity with the AS, and they said, we've got to do some new work, and you guys go try to do this. And so give us a proposal. So that's sort of the process. I was been several years looking for a vehicle that made sense. So it was stumbling around in the dark, I guess.
I say congratulations, because I just find it absolutely intriguing that you found a spacecraft to put on fire, and and it wasn't designed to do that originally. So
yeah, I can add. I remember when David called me and told me that they had this idea about doing a large scale experiment in a unmanned vehicle. It was we referred to it as a garbage truck or a garbage spacecraft, and I just thought it was brilliant. I thought, what a brilliant idea, because up to date, one of the problems with combustion is that the safety issues. So it triggers all kind of red flags. So whenever we have, and I have gone through 5 space flights, and passing the reviews for safety is a big problem. And suddenly here you have an unmanned vehicle that the issue of safety is reduced significantly. Plus, in addition, all the other, because of these constraints of safety, all the experiments in microgravity, they were very small scale. And suddenly you have the possibility of having a large scale combustion or fire experiment in a spacecraft. I thought, David, this is just brilliant. I mean, you have to pursue, and the mechanism to convince the administration is that every single agency have a large scale fire test experiment. The Navy has the Air Force, and NASA didn't. And so this was an incredible opportunity, and really, I agree with you. It was brilliant that David came out with this idea.
Let me ask one question just to change the direction to another area. Now, space flights, especially long duration flights, may require processing of materials in space, be self-propagating synthesis, be via additive manufacturing, welding, joining, or something, which involves all the things that all three of you are discussing. Are we doing experiments, or do we have programs that is leading to those kind of capabilities and know-how and knowledge to accomplish that?
Well, short remark on that. Additive manufacturing is going to be a reality. It is a reality. You've all seen interesting 3D printed plastic parts. They're now moving on to ceramic and metallic parts for very serious engineering applications. However, the world of materials is such that just knowing how to process something in and of itself does not allow you to use it in any commercial or realistic manner, because ultimately it's this long process of qualifying the material, understanding its properties, because each processing method delivers different internal patterns and properties. So it has to go through a long process. I showed you in my talk those 3 examples of turbine blade technology, and that took 40 years before they would allow it to fly in commercial aviation. The first applications were military application, and I think they made that probably prove out for almost 20 years with continuing improvements. Now, the applications for future manned missions will require new kinds of materials, and I mentioned in situ resource recovery. Once again, there'll be aspects of that where we need certain types of data which the processing experiments alone will not give. So there has to be follow-on, and that's one of the concerns I think a lot of materials people have, is that the extent to which reliability can be placed on some of these in situ materials processed in novel ways, particularly under reduced gravity or microgravity, is a high-risk business, because we don't know what those properties are, and we don't know exactly how those materials will respond to the needs.
Well, yeah, as many of these ISRU and material systems involve thermal and reactive processes, so much like a combustion process, and there's been very little work developing them to the point where you could process waste effectively to recover elements from it effectively and reliably in any kind of habitat. And we really need to spend more time on that than we do, even though all the Mars return missions require us to make our return fuel there. So.
Yeah, really, there are not. I think that I agree with David that there are not too many projects. There is a project that actually is a collaboration between NASA and JAXA on studying the flammability of electrical cables. I mean, that's a problem that with time you may have issues of deterioration of the insulation and the materials, even fatigue that can cause cracks in the insulation. The project itself is not addressing the issue, but is looking at aspects of the flammability of insulation in electrical cables.
Well, yes.
Yi Guangjiu from Princeton University on combustion. So, David, you summarized one slide saying that what is the future research in terms of a critical pressure, high pressure, and also a fire safety. So, as we move from the LEO to deep space exploration in the next probably 10 to 20 years, so what would you imagine that new challenge, in addition what you have proposed here, particularly for long duration deep space exploration?
Some of the things that were areas we're interested in, like the supercritical high pressure combustion, is really the terrestrial application. Looking at the exploration applications, I think the waste management, waste reutilization is critical. We expect to probably want to run a supercritical water oxidation reactor to recover more effectively things from the human waste. Is one likely scenario, and other ways to process trash to reutilize it. At short-term missions, even to Mars, you really don't need it, but if you're going any further beyond Mars, the weight to mass value turns out that you want to go to more and more recovery from your closed-loop life system. So that's, I guess, the big area, and also we've got to get better at fire in partial gravity. Right now we have no good place to do partial gravity testing. I showed you the 1G thing. I mean, 0G phenomena. We know that partial G is even more flammable than 1G, but we are than either 1 or 0G, sorry. So the most flammable condition is somewhere around Mars, but we don't know for sure yet. And so, because we don't really have a good test facility, so that's the other thing, big challenge is getting capability to test in partial G.
Okay, with that, I know we are standing between you and lunch. Let's thank the speakers again.
And please reconvene in an hour at, what is it, 1:30, please. Lunch is right outside there. Remember, the people in the committee have blue badges. Make sure you talk to them. And the students in the room, if you wouldn't mind coming up here for a minute, that'd be great. Thank you.
Right.
Oh, I see. It can, but
Well, if you, no.
Okay, let's start getting serious. You've got 1 minute. Is that right? No, you've got no minutes. Oh,
Are you ready?
Yeah, I just wanted to get. Go ahead.
Go right ahead.
Okay. Okay, ready for action? Okay. We're the fluid physics folks, and we're going to try to do the same as the other groups, be on time. We have a very unique panel here. We have Molly Anderson from NASA, a unique choice because she's on the engineering side of what are the true difficulties that are faced for life support systems in general, but other systems too?
Thermal.
Okay, and thermal. So it's a real look at what the real problems are, if there are any. Okay, Mark McCready from Notre Dame, very experienced in this field, interfaces, transport, has played the leadership role in multi-phase phenomena, and you'll hear his input on all aspects of fluids, multi-phase flows, and Greta Tryggvason, also from Notre Dame by chance, and he's been in the doing multi-phase interfacial computations, which are critical for design of future spacecraft. Okay, so but to talk about this, to motivate this, to try to make it sound anything kind of important, like the life science stuff we heard earlier, I want to animate, maybe get this wrong, stereotypes, but we've been in space for years now, right? 100 man years in space. Everything's working out just fine, right? Let's go to Mars. No problem. Right? No problem. The fluids problems are not a problem, but the fluids problems have to do with the fuels. 80,000 pound tank to space is a liquid. It's a fluid mechanics issue. Self-gravitating globular thing, weird wedding thing, weird stuff. If that's not done right, we can't afford the mission. It's a pretty heavy. It controls the CG of the spacecraft, maybe. Okay, but it's also the small stuff, the life support stuff. If people are going, stuff's got to work. The toilet's got to flush. If it doesn't, the crew dies. Now, there's all kinds of fluid systems in spacecraft. For the life support stuff, it's habitats, water delivery to plants, plant and animal habitats, that kind of thing. It's the toilet, right? But it's also the condensing heat exchanger. It's the CO2 scrubber. It's the astronaut backpack It's got everything. It's a space station too. Okay, so it's in every aspect, and if it's not right, we got problems. Now, a lot of crew time is spent maintaining the system. Yes, the space station is incredible that it's been a life support, delivering life support, right, for this many years to that many people. But it's been working because of up mass. It's been, spares have been used. We continue to resupply it, and we freshen it up with fresh water, that kind of thing. So we got to close. We got lots of fluids challenge. We got lots of challenges with fluid systems because they've got to be robust. When we have to use, and this is an input here just for kicks, to use this time in the space station to prove out technologies that can make the systems more robust and buy down the risk. Because if the life support systems relating to fluids go bad, then we got issues. So you could be from industry and say, let's go, we can do it, we know how to do it. You could be a numerical person, you can say, compute it. You don't have to measure it. Okay, you could be an analyst who says, there's lots of research that we could do. And so far, there's been a lot of neat, cool, beautiful research been done in fluids, liquids, and all kinds of fluid stuff on the space station. But what's the right way to go ahead, go forward with this? And this is something that the panel or the board really would like to know. What is necessary? Where are we now in fluids after so many years of research up there? So we have a unique panel to address those things, and let's just get started. Mine? Okay, sorry.
There we go. So my name is Molly Anderson. I serve, as all of us do, a couple of jobs at NASA. I am a principal technologist for the Space Technology Mission Directorate, working on life support and thermal and a range of technologies. On that end of my job, I am supposed to be not only open to, but looking for new technologies, new ways of solving our functional problems based on new discoveries in science and new methods. But my job also spans all the way to the other side. I'm deputy for our life support and environmental monitoring system maturation team, which is the working group, whatever, charged with making sure that we are ready to go to Mars. And so the first kind of question that we got on our list was, do we know enough to go? I don't know how many science committees pay attention to engineering committees, but in the last couple of weeks, Bill Gerstenmaier from the Human Explorations side of NASA just quietly put some charts out to the NASA Advisory Council for when a Mars habitat would be launched, and it actually had a date on it of about 10 years away, which is kind of terrifying for me, since I feel like we've been working on Orion for about 10 years, and much of that is budget, but still. So you say, are we ready? I say, kind of yes, kind of no. I think we believe, from a system point of view, that we have at least one credible way of doing all of our functions. So there's not like an imagination gap in figuring out how we're going to get to Mars. My concern is much more in our experience of how many tries it took to get things right on the space station, and these timelines that are being put in front of us, and that we really would like to achieve. As usual, are success oriented, and so when we talk about, are we ready? What mission are we talking about? Just to make sure we're all on the same page. The human exploration side is looking at putting up vehicles and modules in a lunar orbit and leaving them there, having the crew visit periodically. So that does interesting things in terms of sometimes systems are on, sometimes systems are off, and transitioning between states, which I think is important. And then a Mars habitat, and all your Mars pieces are delivered either to the lunar orbit or straight to the Mars surface. And then the crew gets there and turns them on and rides in them. But that means that while we will have logistics to get to like a lunar orbit to leave, that's pretty much going to be taken up by food. We don't have mass time, crew time, whatever, to go and re-outfit the entire fluid system because we did something wrong in our design. So those kinds of things are what scare me. We have a number of let's see. We have a number of challenges that are going to be different for these future missions. Like I said, dormancy is one that is starting to really bother our community. People are asking, that we basically have two models. The space station has been permanently crew tended, permanently cared for. The space shuttle was returned to Earth after short missions and completely, redone and cleaned out. And so now we get neither of those. We get a fluid system that stays on for a long period of time. We have a lot of sort of classic problems of unexpected fluid transfer that in retrospect would have been obvious, but you have jumpers, where you have porous, lines, where you have porous hoses, where you have quick disconnects because you want your systems to be very maintainable. Those all look like places where water is going to evaporate out of a system, and now you have a pressure drop and a bubble where you didn't expect it. You have a bubble trapped in QDs in a whole lot more places in the system because you decided you wanted it to be maintainable and to think about those follow-on effects. So I think there's and then plenty of interfaces, as we were talking, plenty of interfaces between the fluid system and the biological system. So to me, they're all linked problems. The whole people have said, well, can we drain and purge the system and refill it with water? And that's for the sake of biological control, right? Or people have said, can I bring the fluid temperature way down for the sake of biological control? But is that going to do anything else to properties? We also potentially have components in this system that operate both in microgravity and in a partial gravity. So that's a dynamic and a spectrum. It's not a stable, consistent design point. So the next question we got is, what are the most critical subfields of research required? And can they be prioritized? So I'm interested in a lot of the topics here today, but sticking with fluids, my concerns are I think there's from the point of an engineer, there's a brilliant science workforce out there that's studied a lot of really interesting systems. But my systems are very imperfect. They tend to have varying fluid properties depending on what they're made of day by day, and so their behavior may not be consistent. I have a lot of failures that seem to be based, when we look at multi-phase flow, it's actually solids. A lot of our failures are based on particulates somewhere in the system, either carried by a gas or carried by a liquid, or precipitated, created because of some unexpected liquid conditions, something like that, something allowed to stay in one place for too long and start to precipitate. I'm also worried about things failing over time. So you're talking about separators and condensing heat exchangers. We have we have sensitive components, and we have a lot of microgravity fluid ideas on how to create passive systems, but I don't have a lot of time, and I don't think an engineering industry can afford a lot of trial and error design. Here, let me just spend 5 ecosystems and see which one works. So, I mean, I look at this from NASA, but also trying to assist commercial space. We need to be able to translate the science into effective design tools so that we at least get things right on the second try, if not the first. So from a numerical models point of view, there are great mathematical models. My concerns are how do we make them accessible? How do we handbook is probably maybe going too far. Engineers want to just be able to look things up and say and pick a number off a table, but how do we get enough knowledge out into the field that we know when it is most important to use the detailed mathematical models? What models can we use to easily predict that we're likely to be in trouble and then spend the time there, because our systems are too large, I think, to model the entire thing at the level of detail of numeric codes that is accurate. Right. And the world, like I said, the world of life support so far has just basically tried to avoid multi-phase. It's a scary thing, and you try. If it's liquid, keep it liquid. If it's gas, keep it gas. And our problems happen when something unexpectedly is not what we wanted. And the last question we got was what's the ratio of fundamental research to applied research? And I was thinking about this very, very differently. There's great discovery that comes out, but also I look at when I'm funding something, it seems like dollars spent on research is directly equivalent to graduate students funded. And then I look at what happens to those graduate students when they come out, and I think most of them go into industry as opposed to academia. So I'm very flexible about what kind of research they do, because I want us to have a workforce full of skill sets that can predict, analyze, and cope with these problems. That tends to be sort of a question of complete education and less about, not always about their particular thesis, but about the way the field is, I guess, taught and disseminated. So there's kind of my summary of highlight points, and I guess we'll take questions later.
So hopefully my slides will pop up here. I can't give a talk without slides or chalk. Does that work? Okay, so that title's probably fairly irrelevant, but I was thinking of some of the systems that must be important to have multi-phase flows in them. We've been skipping over this slide all day, so I'm as older than I'm at least as old as I look. Here's some. I hate to think that way. Here's just some of the problems we worked on over the years: bubbly flows, gas liquid flows, and solid liquid systems for reaction. Waves. I did a lot of work on waves. This experiment here was actually one of the things we were mimicking microgravity by matching the density of two different liquids, and so that was a vertical interface with waves that formed this way when the thing was rotating. So you can do all kinds of things, and this is the point of that is there's many things you can do on Earth that can mimic things that have microgravity, and they cost way less. And then just some other work, and we've done also molecular dynamics simulations of trying to figure out nuclear boiling and transport properties in ionic liquids as well. So we kind of covered the thing, but mostly I did experiments, and when I've interfaced with the other people in the two-phase flow community, there really were two camps. There were the experimental people and the numerical people, and we used to fight it out pretty well, and so I think it made both of the groups stronger, so that the people who do computation had to prove it, and I think over the years they have, and so I think that that's really advanced the field quite a bit, so that you can calculate things now that you really can't measure all that well. Oh, wow. Okay, here we go. Anyway, so looking at going to Mars, I'm expecting that whatever systems we're talking about here are going to have to be designed right on the edge, which I'm sure scares the people here. But also, and so they're going to have to use multi-phase flows to get maximum performance, but right from the beginning, rather than having to design out a bubble that got in there. You want to know what's going to be there. So the first thing is that we're going to, you're going to use those systems, but we'll know it from the beginning, and I think that's an advantage over where you are. But almost for sure that you're going to have to answer questions we haven't answered before. Okay, so you're going to face that point where you're going to have to make a decision. You're the engineer. I believe it. I'm confident, and I'll bet my career that we're not going to kill the astronauts. That question's going to happen, no matter what we do. Yeah. Well, okay, but that's the thing. When you actually get to make the decision, yeah. No, the point is that this. So that's but that's what's going to happen, because we're never going to know anything. So what I'm going to do is I'm going to show you some flow situations, but I'm pretty confident that if you could define the flow situation, we can calculate what happens there. We can tell you what's going to happen there. So it was, from a scenario standpoint, when you go to design a device, starting at the beginning and putting pieces on and getting it bigger, each of the little problems, most likely you know the answer to. It's just at some point you reach the system level problem, and that's gets harder. But every little piece we probably know the answer to. Okay. Right. The thing that happens at the system level is even if you have each of the little pieces correct, there is this accumulation of small errors and dynamic uncertainties. So as a steady state pressure, I know the answer. What if the pressure upside is bigger than I think? Those kind of things happen. But still, if you have to build it and test it, it's going to be hugely expensive. And so I don't. You can say we want to do some more experiments, but the thing that you're going to have to build an experiment on is a very big system, and that was very expensive, and you may not be able to have the payload to get it there. So you're going to be back to making that decision. I can either believe it or not. And I'm going to bet my career on keeping the astronauts alive. But anyway, the engineering exists. The thing that you've got going for is that there are people who've flown these experiments in the 90s and early 2000s, and engineers at NASA have this experience right now of doing this. And so to mitigate some of the uncertainties and the unknowns, it's the current engineers who have first-hand experience with making these experiments work in the 22nd trajectories and other places. And if they don't, if we don't get to use them now, they're going to retire. So you've got to get this. Has got to go now if we're going to do this. Otherwise, you're going to have to restart a whole bunch of things. So anyway, so here's. I just want to point out the kind of problem that we're talking about. This is John McQuillan's. You can still go to this website and look at these videos. These were really cleverly done. They showed us sort of all the stuff that can occur in multi-phase flows in microgravity. And so that scoped out the problem, and so that knowledge is known. And again, you got to keep it. McQuillan won't stay forever. And if you break them down, they all look. You can break multi-phase flow into these sort of classic problems where you have this kind of different geometries. And the reason this matters is if you do some calculations of the pressure drop for the dispersed, which is all mixed up, versus a stratified, which is two separate flows. The pressure drop ratio can be a factor of 30 difference. And so that matters, 30 matters. Or if I have this situation that John's movie here where he was, the flow was coming up here and it could split there and there. If you do scenarios, you can calculate the range of possible splitting flows here, but it's not trivial. It could be, again, all the liquid could go one way, all the gas could go the other way, or the other way around. So it matters, and so we have to get these right. But to a large extent, I think you can. So if I look at this flow configurations, I'm going to draw some cartoons here. So here's some of the classic cartoons that if you would start with, if we can get these right, we can pass test one. And so here's the classic T problem where you have a flow coming in, it's a mixture of gases and liquids, and you want to know how much gas and liquid goes there and how much gas and liquid goes there. And, so the question is, can you solve that one? Well, we'll ask our esteemed colleague. Same thing here. I would just a side branch, similar kind of thing, but you care about how much gas, how much liquid, and it could be that the pressures and are changing as a function of time and those things. The tank problem, you would want to be able to get this one right. So I would want to get knowing what's coming into my tank, then where does the liquid go? And so whether I'm venting the gas here or not, or whether I'm venting liquid here, it's possible, many configurations exist, and then if you say I want to get the liquid out later, how do I do that? So this is the kind of challenge, and this is also the computational question we would like to know the answer to. One of the ones that everyone complains about, a bubble getting stuck, it sort of looks a lot, or a lump of fluid getting stuck looks like this. And so you have a situation where liquid is somehow here, and you get a bypass region. So whether it's because that's it's either you intentionally have this surface chemistry different, or you have some crud here. Crud's one of my favorite technical terms. That you liquid gets stuck there, and then can you get it off? Again, that's a computational problem that could be addressed, or anywhere else in your whole device, just because you've got a lump of fluid and everything else is flowing greatly, but it's fine, but you've got a lump of fluid. Can something get stuck there? How big would the thing be? And then can you clear it out? And so there's another question you could. So any of these things, if you can ask, if you can conceive of the situation, then you can ask the question, what would it take to solve the problem? And so I think you could probably do that. Another one would be the stationary stock bubble, right? So if you area, there's a huge pressure difference across this interface, and then the answer is you can't get it out. Sorry. So if that kills the crew, then that was a bad design. Okay. So, but nevertheless, again, it's a knowable kind of flow geometry. You can change the chemistry depending on whether you think you have different contaminants in here, but at the end of the day, it's F equals ma. We should be able to solve F equals ma. Another one here, the heat transfer problem, where you've decided we do have a heat transfer surface. We've got to put heat flux into our fluid. If it's a dried out region versus a wetted region, that's kind of important to know. It can be dealt with, but you need to know that, so you need to know where the liquids and gases are. One of the ones that's flying right now, fortunately, is the packed bed problem, because it's very, very difficult to figure out how much water or liquid would be in the packed bed. What the holdup is, but I might like a packed bed reactor both for high contacting area or for catalytic process where I'm doing reactions. It would be it's really important to know that. So this is, fortunately, this is flying now, right? Yeah. So this was one that we'll have new experiments on. But in any case, these situations are sort of knowable kinds of things, and you can, if you want to, take the computational approach, and to do it is you conceive of the situation, and then you can do scenarios of calculations. What if, and then bracket the whole performance of the system. And so with exact numbers, I think you can get to this. And so the real challenge that you might want to pose is just, we really need a heat transfer surface at a certain heat flux. Can we do it? Well, challenge us. Thanks.
Well,
there you go.
Okay, thank you so much. So I'm going to talk a little bit about computational modeling. My main message is going to be that we have really come a long way in the last decade and a half in computing multi-phase flows, and we are actually facing now new challenges, or rather we are having opportunities to face new challenges that are going to change the directions of our research in the future. So this is a busy slide. The main takeaway from this is that I've been in this business for a long time. I've been working on both development and applications of numerical methods. Maybe the noteworthy thing is that I was the editor editor-in-chief for Journal of Computational Physics for a long time, and so I really had an opportunity to see the development of various numerical methods, probably more than you really would want to do. So, in single-phase flows, there's been a lot of progress made, and those methods are now applied routinely to design of airplanes, cars, even engines. And in multi-phase flow, even though the origin of efforts to simulate multi-phase flow go all the way back to the beginning of CFD at Los Alamos in the late 50s and early 60s, those methods lag significantly behind what is possible for single-phase flows, and it's only really in the last decade, perhaps, that similar progress is being made. So we're far behind in what can be done there, and one of the challenges is to bring it up to sort of a compatible state. Now, obviously, as it says on the slide, the big difference in microgravity is that free surface flows and multi-phase flow really matter. You want to use such flows because they have better heat transfer properties and various other benefits, but they're much more challenging to work with. So one of the things that I've been involved in, and I think has been one of the big progress that has been made, as I say, over the last decade or so, is our ability to conduct fully resolved simulations. This compares with direct numerical simulations of turbulent flow, which really have been instrumental in development of large eddy simulations and so on. And we're getting to that point here. So there are many methods currently available. When I give these talks, people ask me, or at least used to ask me, what method is best. I just told them that it was mine to make the answer as simple as possible. But the reality is that there are lots of methods that can do very fine job at simulating multi-phase flows by now. And we have applied these methods to a large range of problems, and in many cases we're doing quite well. I have a rather condensed list there at the bottom of this slide, but maybe this being NASA, it's more appropriate to show pictures. So here's a sample of many things that we can do. We can do, certainly we can do clean bubbles and drops in turbulent flow. And we have done simulations of hundreds of such bubbles and evaluated things like effect of heat transfer. One of my favorite is down there. We did some work for DARPA a long time ago, where we really found that subtle deformability of bubbles was critical for drag reduction, and was actually one of the things that was later confirmed by experiment. And I come from a generation, I don't look as old as Mark, but you know, I'm just about as old. So I come from a time, you know, when we could sort of post-dig things. So this was a prediction, which was a new thing for me. But anyway, you know, there are simulations, there are thermocapillary migration, boiling, electrohydrodynamics, and various other examples. So the bottom line is that for many problems, we can actually do quite well. These kinds of problems are problems basically where we know and trust the equations. So if you can give me equations, I can simulate it fairly well for a fairly large range of scales. Now, of course, the key challenge is that I cannot, my simulations cannot give you physics that I didn't put in it. Or as somebody said when I gave a talk, at most it can give me, simulation can give me the physics that I put in. So certainly complex physics remains, you know, a challenge for these kinds of simulations, and that's where we need experiments. We also have a lot of circumstances where there really is a large uncertainty of the setup, the boundary condition, boiling on foul surfaces or even pattern surfaces. And of course, there are lots of problems with multiple scales. So multiple scales come about in many different ways. They can be just because that's what the flow regime does, and also because of physics. So roughly speaking, I've used this classification of multi-scale problems for a long time. It actually was suggested a long time ago by Wine and E and Bernd Anquist. But basically, you can look at multi-scale problems sort of in two ways. Suppose you have a simulation where there's a dominant length scale for bubbles and drops in multi-phase flows in general. This is usually set by surface tension, balanced with viscous forces or inertia. So there's a dominant length scale that sets the resolution you like to simulate that. Then you have two multi-scale problems. One is, and this is sort of the classic turbulence problem, you want to upscale. You want the simulations to help you develop reduced order descriptions of the larger scale flows. For lots of problems that I deal with, this is the main problem. We want to do really large scale industrial plant. We really have to go to Reynolds numbers and scale ranges that we cannot possibly simulate, so we need to extract these average models. And a lot is going on in this area. One of the very exciting developments in this area, motivated in part what people are doing in single phase turbulence, is to actually try to use machine learning techniques to correlate these unknowns, the closure terms, with the larger scale flows. For many of the systems that are relevant to NASA, it's sort of the other multi-scale problem. These isolated region where something happens that takes its place on a much smaller scale than anything else in the simulations. This could be very thin films or threads in multi-phase flow due to the breakup of drops, but it could also be due to a different physics. Physics that takes place on a very different scale. Obviously, in combustion, things take place on a much faster scale than anything else. In our kinds of problems, for example, mass transfer is an example of scales that are much smaller. The ratio of mass diffusion to momentum diffusion in liquid is often in the range of hundreds to thousands, and this leads to very small scales. So these multi-scale problems certainly pose a challenge for us. So let me sort of move on, because I think I saw the 5-minute sign just a minute ago. Sounds like we're being ready for the 2-minute sign. But the point is, again, as I started by saying, is that for a very large range of problems, we're actually doing very well. Okay? There certainly are issues that still have to be resolved. And of course, as an academician, I'm happy about that, because as you know, in academics, we really only like to work on problems. We actually don't really want to solve them. But the practical aspect is that we really are run into a situation where we have capability academically which really need to make their way into the practitioner community of practitioners. Now, some of that is already taking place. Certainly, Fluent has volume of Fluent, OpenFOAM has similar capabilities. There are specialized codes like Jerry's and Basilisk and so on. So some of it is already happening, but definitely not to the same degree as we can use routinely large eddy simulations in turbulent flow. The last point is sort of something that I have gotten more concerned with as I get older, probably, I should say, or maybe more senior, and that is how you lower the barrier for entry for new people into the field. In many ways, we had it good because the problems were relatively easy and we took them, so the problems left for our students are the hard problems. And I think it is sort of on us to try to make it as easy for them to enter the field as possible, and one suggestion was there at the bottom of the slide. So again, the summary basically is that you know, we have come a long way. There are some challenges left, but I believe strongly that the availability of these computational methods is really going to have a profound impact on both how we deal with sort of routine designs, where we can use some of these techniques to develop optimum geometries and so on, but also novel things. There are lots of things happening, for example, with pattern surfaces. You know, the idea of heat transfer by jumping drops and so on. You know, you certainly can think of functionalized particles that transfer and enhance heat transfer, just like the red blood cells enhance mass transfer and so on. And I strongly believe, as I say, that this is going to lead to profound changes, but we really are to the point where now lots of new opportunities are opening up, and how to get this into the hand of the practitioners is one of them.
Is it okay?
Okay, before we open questions, let me also just take a few minutes to ask my own. And I don't know who to start with, but in fact, I'm thinking about what you're saying. We know that as it is right now, that a lot of fluid systems are problematic. I am also buoyed up by the things that you say. Okay, so I agree, and I'm impressed too with those kind of results that show that, hey, things are working, we can do this, and there's not a need for a bunch of more experimentation in fluids in space. But we need a slew of creative ideas that can overcome the unexpected phenomena, so that our systems aren't so much dependent on this coating on a surface that might flake off in time due to cooling and dry out events that then destroys the system. Okay, there's a condensing heat exchanger, for instance, that relies on a coating. The coating is flaked off. The wetting now is completely transitions from a film coating to a slug-like thing, and it's ejecting liquid into the cabinet. Okay, that's a failure that's not anticipated. How can our knowledge and our growing confidence and our ability to analyze things improve on the design thing, so the next round can be more insensitive to things like that? I don't know who to ask. Who wants to answer? Can you? You? Mark. Hit the button. Yeah. Start
no, I mean, part of the problem is that I don't personally know I've talked to NASA people, I've interacted at conferences and workshops, but no one's ever come out and said, here's exactly what we need to do. Here's the one we've got now, and here's what doesn't really work about it. So part of it would just simply engage with some fraction of the academic or other community that you think is best, and get it started. The other way to do it would be to have prizes, right? You would publish the specifications, you would give away a prize for a winner, and then you would just take all the ideas and then, incorporate them into a real design. But if you think you need new designs, you need new people. But in either case, at the end of the day, the new design is going to need computation that needs to be done exactly right. And as Greta says, if it gets to that point, then the question can be answered.
Is industry currently capable of either making those connections and doing that work? Oh, if you had something to say, you better say.
Yeah. I know there are small businesses out there that are proposing new ideas using things like capillary instead of our conventional methods, right? But if they make too many attempts and fail, our attention span is only so long. Let's be honest, right? And even if the concept is not flawed enough, black eyes and attempts, and people don't want to be the one who picks it back up again. I think we have, just because of the way the money flows, we end up going to the small business community a lot, looking for ideas, but there is the STTR, which is supposed to be a combination of business and academia, and I don't know if it's utilized well enough. I don't know if I feel like businesses have an idea sometimes, and then they try and grab somebody from academia who can help them. I don't see a lot of push from academia up through. Is there something you can do with this idea? From my perception, I don't know what it looks like from the other end.
I think it is fair that the key to get those capabilities out into the hands of industry is educating the right people to do so. And there are a number of programs at your university emerging where the focus is on sort of on computing and science and engineering. I don't know. Indeed, I would say that the focus in many ways is not on industrial usage. Okay, so there's a lot of programs that train people for research and so on in those areas. But I think what is needed is more focus on people who can actually do something with methods that are there but maybe state of the art. The thrust towards bringing computational techniques into the hands of particularly smaller manufacturers is certainly well understood. You know, for reasons that I don't know, I get sort of a European newsletter and something like this every month, and there's a lot of discussion there. They certainly have all the right words. But even in the United States, in the latest, what was it called? There was the manufacturing bill. What was it called? The American Reauthorization bill. Right. So there was a statement that literally said that Congress finds that. I don't know if that's a good thing or not. That, you know, that it was extremely important to find ways to enable smaller suppliers to use the same kind of computational modeling that is now commonly used by sort of the larger providers. Okay, and I think that is a sentiment that is widely shared, and certainly I think we in education appreciate that. That's an important step to take.
One more. Okay, so how about the space station? Pretend 7-year life. That's what we got. Is there a set of benchmark experiments that would be ideally suited to provide useful guidance for design and maybe benchmarks for numerics and analysis, analytic models? So that would be helpful to the engineering community. That would convince people, for instance, that may not be otherwise convinced. All of you could answer.
We're going to attempt to test our best guess of what the systems are. It's often frustrating because on space station for an engineering system, you see success or failure, and you don't get insight into the system as built. It's made of metal, not clear things. It has fewer sensors because your telemetry limited, and so we get a pass-fail, but not necessarily a deep understanding.
Well, we had a workshop on this a year and a half ago, trying to come up with such experiments, and the limitations of the fluids rack, the limitations of the payload left us with not all that many, not much of a range of things you could study, because what you'd like to study is bigger, interconnected, and the kinds of things that would help with the systems. And that wasn't really what was possible given the expected payloads and the expected time scale for space stations. So we proposed things basically that came off of the pack bed that was already there inside the glove box, and replacing the pack bed with some other geometries, just trying to get some other geometries, but at some point, you know, if it's you're not going to fly a dirty fluid because you don't know what it is. Even all your fluids are dirty. So we're not going to propose a set of experiments with things we don't know just to find out what's going to happen, because it wouldn't generalize. And if it's a clean fluid, we can compute it. So the forensic discovery on things that had already been done could be way more valuable than trying to propose, again, something that fits this size geometry that we can sort of see into. We have the pipe sizes of a centimeter or a little bit more. And even if we make them triangles, it's not going to dramatically change what we learn. So it's hard to see how that's going to, the space station capabilities are going to dramatically change the picture of having to make a really big bet on a really hard problem to, you know, keep these guys alive and to Mars.
Yeah, I think I want to second that. The experiments that are needed are either on physics that we don't know. Some of that we could do on Earth, but we should focus on, as I say, discovering physics that needs to be modeled and go into models. And of course, the other experiments are simply exploratory experiments where we go and look and see, you know, what kind of phenomena could happen that, and those usually take place on a very large range of scales that may not fit into the current facilities. For clean multi-phase flows governed by the standard equations for fluid dynamics, you know, we can solve that on the computer, and we don't need an experiment to prove that the Navier-Stokes equations describe well clean fluids. That's pretty much done. Okay, so the kinds of experiment that we would be interested in really has to be driven, I think, be sort of by well-defined questions, not just because they fit in the current box. So.
Okay. Let's do it. Find your way to the mic or grab it.
Yeah.
Throw the box.
I have the box. Okay, I asked Swami from University of Pennsylvania. I was very surprised that the panel didn't speak about porous media at all, because in fact, particularly in reference to heat transfer, the weak structure of looped heat pipes, which are the ones that are used, essentially have a serious problem, because I have worked on that problem in conjunction with radiators, the reactors and so on. Because in the loop heat pipe system, the wick structure and where the fluid is in outer space, and how does this capillary flows, how do they get affected, significantly will affect the heat transfer. So in essence, you will have no control over cooling or heating if you did not know porous media heat transfer. And I thought the panel just missed or glossed over it because I didn't see it at all in the discussion, although you did mention, Dr. McCready did mention something in heater associated with some kind of a cartoon.
I mean, in 10 minutes, we can't conquer every example. I agree with you that there are a lot of challenges there, and I would say that right now, the applications for community for human missions is not excited about adopting them because it could fail.
Matt Colquitt from ASGSR. My question is about partial gravity. I hear initially about the criticality of fluids to basically a lot of systems on these space flight systems, and particularly for life support. I hear we can do a lot of modeling, and I think I have a pretty good understanding of that, and I think we're a lot better now nowadays than we were when we first put up the space station. But still, I also hear what Dr. Anderson spoke about when she said, hey, look, when we put up the space station, we got a lot of surprises. Fortunately, we were in a situation where we could make changes because we were close enough to Earth, and you can send up another replacement. That's not going to be possible on Mars. That's the reality of it. So I'm sitting here thinking that somebody's got to be doing some testing under partial gravity. I don't think it's going to be as severe as was cited in the combustion environment, but there's probably no doubt that there's going to be potentially some surprises. So I'm just mentally thinking about the environment that you have to do partial gravity experiments. You've got the centrifugal unit up on the space station. I'm not sure it's large enough, but I guess I was also wondering about the Sapphire experiments that the combustion group was talking about, where you might be able to not only get a larger system in there and look at microgravity environments, this companion to what he's been doing, but actually some of the capabilities there. And I don't even know if this is possible, but if you could have a flight trajectory that adds a thrust to the vehicle during its descent, so that you have a constant, like 0.3 G effect or something like that, pick a number, but actually it changes trajectory on its descent to have a microgravity environment and do some tests on that. Just throwing it out, and I don't know whether you have any plans for this, but it seems like it's a missing element of anything that I heard talked about here.
Then light it on fire.
So I'd like to follow up on that comment just a little bit and ask a nuance of potentially the question. In all these simulations, and this is not my area, so please forgive any naivete. When you simulated zero gravity versus when you simulated at one gravity, what does the shape of the curve look like if you vary between there? In other words, is this a normal thing that you can do by changing the value of G? Or is it a normal part of your simulations? Is it a difficult part of the simulation?
So on the computer, you can set gravity to anything you want. A long time ago, I told a graduate student to just turn off gravity. My door was open, and there was an experimental colleague outside, so he sort of looked in with a funny face. But you know, as I say, on the computer you can set it to anything you want. Indeed, in many calculations you work in some normalized form that goes into some non-dimensional numbers, and then you look at the whole range. So if we have the correct model, seeing what happens in microgravity or even reduced gravity is not complicated.
Okay, if it's not complicated, is the shape of the curve a straight line? Are there steps, or are there exponential effects?
In most cases, I think it is fair to say that while the things change significantly once you turn gravity on, particularly for a long time, but off or on, that's a big change. Okay, but once you turn it on, often it is simply a matter of time scale.
Thank you.
Often.
Yeah, you can see that we've done experiments with different ratios of density ratio and things like that in gravity, and the rates change, the qualitative things stay essentially the same. Quantitatively, you get a little bit bigger, faster growth rate, or the shape of the interface would be a little bit different. But there's I've never seen a complete surprise as I cross that threshold and show there's nothing in the equations that suggests a complete surprise. If you turn it off completely, you may get a singularity, but once it's there, it's pretty smooth. So I wouldn't really anticipate a lot of problems.
And if you have all the right physics in the equations, the things work out. But if you forgot to have some gas diffusion thing in there and some thermocapillary thing takes over, then you got issues, right?
I'm Mike Bickel, Loma University, also ASGSR board member. I want to bring it back to what you started off the conversation about. With one of the fluid problems we have to deal with is waste management and life support. A lot of those, by definition, involve biological systems. So I'm wondering how well your models are incorporating things like biofilms and that kind of fluids. Are you say you can model everything, but can you really?
The question of biological fouling, right? At some point, you need to translate the biofilm into a constitutive relationship between stress and strain. And that experiment could be well, if the colony is completely different in microgravity, then you would have to get a microgravity colony. But to the extent that it's just something you could grow on Earth, once you do that, you get the constitutive equation, and which would be straightforward for a biofilm. It could be incorporated at that point. You just have to know that it's there.
Sure, but the biofilm isn't constant. It's constantly changing.
But that's not a problem. We can put a dynamic reaction term in that would account for that. Because the kinetics are not that complicated. I mean, the kinetics can be modeled as something that's not that complicated. So it's just a matter of knowing that there has to be something there, and then doing the right rheology experiment, basically, to incorporate a biofilm or something like that. And even breaking it up wouldn't be a problem. But if you don't know what's there, then that's a really big problem. But as far as modeling it, once you've decided we know there's bugs there and they're going to make biofilms, and we have a polysaccharide layer.
Maybe I can add to that a little bit. So a lot of effort has been spent in the last decade or so on quantifying uncertainty and propagating uncertainty through the equations. So equations with uncertain boundary condition and so on. So for things like surface finish or biofilms or fouling and things like that, it's likely that there's going to be considerable uncertainty in your knowledge. You need a model, as Mark says, but the model may have significant error bars. And in the multi-phase flow area, we haven't done nearly as much as has been done in single-phase flows, but it's an area that is emerging, and I think, in a decade or so, we're going to have a pretty good handle on how uncertainties in properties of surfaces, for example, or material properties, are going to propagate into uncertainties of the prediction.
Thank you. Yeah, my name is Jeff Smith. I'm from the ASGSR Society and also Kennedy Space Center. And I have a concern about something that Molly Anderson just touched on, and then the comment later about clean fluids and dirty fluids, and I'm concerned about the situation where a multi-phase fluid is doing something unexpected when one of the phases is a solid phase, a particular contamination of some kind. And I'd say NASA has been flying dirty fluids for quite some time on the ISS with some unexpected and pretty interesting results. Particularly, I'm thinking of things like the urine reclamation system and calcium carbonate type deposits, and biofilms was just mentioned as another potentially particulate that could get into a system and cause great surprises, and then propellant systems and propellant handling systems are extremely sensitive to particulate contaminants, and when you're trying to do something like handling propellants on Mars or on the Moon, you're going to get potentially contaminants. So I'm concerned and curious about the ability of the computational methods to address concerns that might come up where we're looking at particulates and contaminations and dirty fluids, and help NASA prepare to design systems that are going to be robust enough to handle these dirty fluids.
Well, as Mark said, if we know it is there, the probability we can predict it is significant. The challenge is, as I said, also, you're not going to get any physics out of your equations that you didn't put in. So it is really a modeling issue. Modeling meaning sort of modeling of a physical systems. I tend to think that we have made significant progress in some parts of this sort of the challenge, and hopefully if we integrate what we already know, we reduce at least the uncertainty. Certainly people have seen that in other applications of CFD that, a computational model doesn't totally alleviate your need for an experiment, but it certainly reduces them quite a bit. Okay. Now, as we get better and as you find modeling become more of an integrated part, hopefully we would find ways to integrate these models for more complex processes into those. But can I predict how the urine reclamation system fails right now? Probably not.
Thank you.
Hello, I'm Stephen Collicat. I'm here with ASGSR, and I'm a professor at Purdue University. This being an opportunity for public input, I'm not asking you a question, but Mark Weislow was concerned about the rightfully, I think, the 7 years. Can the current schedule remaining for station, and what are we going to do? We've heard from Molly about, is something going to work? Is something going to fail? How do we know? We've heard from Mark McCready about, I think, a very, very wise comment about the people who were doing these parabolic flight experiments and others in the early days are getting old. Where's our new community? And Professor Tryggvason has spoken repeatedly about, well, if you have a good model, we can compute. And all these things, I want to point out my input to committee is very much complementary to ISS, and to get a payload or two into ISS puts you in a very, very unique group, and there's many, many other researchers, far more who can't do that. And that is the commercial suborbital industry now providing 3 to 4 minutes of high quality weightlessness. It's not 20 seconds of bouncing around like we enjoy in parabolic flight. It's much higher quality. Blue Origin's flying, Maston's flying hundreds of flights. Virgin Galactic's in flight test. Exos is coming back into flight test. So my input to the committee is and this industry was just budding when the decadal plan was created. So it's a very timely addition, I think, that please do consider the great capabilities of this new industry and how those complement what's in space station and parabolic flight and drop towers. Thank you.
Dan Dumbacher from the committee. I want to maybe go off on
the state of the art and what we need to know and what we don't know in order to do things like propellant depots and some of these other ideas that people have put out.
Take this one.
So I mean, NASA is investing right now in simulation of cryofluid management, and we're doing small-scale system partnering ISS experiments with modeling to try and validate them on a small scale. I think it comes down to that optimization question and the question of how many tries does it take you to get it right. You know, I think that we have ideas that will work, and whether we get the design right the first time will depend, you know, on how well quantifiably validated, you know, really our design tools and simulations are. The mental model is generally correct, but those knowing how much ullage you have left in your tank, or you know, is the difference between making it home or not, and for depots and things like that, right? Effectively making use of your depot and not just filling your tank up half every time is important to profit margin. And you know, when you look at other industries, right? They're making tiny improvements to improve profit margins because they have huge scales. If we want to get to that kind of operational confidence, I think we can make something that works, but if we want to really become an industry, we need to make it better.
Hi, Cheryl Nickerson from ASU. I'm not a physicist, so let me just state that up front. But to me, I see a beautiful opportunity to continue the collaborations that NASA has really just started to invest between the physics community and the biology community. And I'm going to follow up on what my colleagues said here on biofilms. We know biofilms can clog anything that's got fluid running through it. We also know that biofilms are forming on the ISS. They have clogged water systems before. No water, the crew dies. All right. We also know that biofilms are extremely hard to clear with disinfectants, and your body's immune system simply cannot clear them, and they're responsible for about 70% of all infectious diseases that we get. So we also know that biofilms have been reported to form unique architectures in flight that we don't get on ground, that could suggest they may not be as treatable with the conventional disinfectants that we like to use up there in the water system. Silver on the Russian side, correct me if I'm wrong, I think in the future
balance is going to be going to silver. Right now they've got iodine. So I'm thinking there could be, and I should also mention that while biofilms are forming and systems on orbit, we don't really know what they look like because it's not transparent to see them. And so how can we build into your model? There's a lot of groups now that are doing biofilms. Does that present an opportunity to enhance the biologist's ability to work with you to understand fundamentally how that changes, has the biology changes, how that changes the trajectories and the models that you need for these kind of operation systems, bioregenerative systems?
Well, I'm not a biologist, so but I believe that there would be significant opportunities. There have been some effort in sort of my general community to model biofilms, but I don't know much about the details. So I can only sort of, declare a general interest or general believe that this is an opportunity, but that's probably as far as I can go with sort of what I know about that specific thing. So.
Thank you.
Yeah, I think that's true, and there have been a few, there are a few papers in the Journal of Computational Physics that I came across in that area, but I don't think, I think there's still lots of opportunities there.
Okay, I want to thank the speakers very much for their input. Thank you very much for the good questions, and we'll move on.
all here? Yes. Two minutes to go? Okay. Two people.
Oh, yeah, I have 2 minutes.
Oh, okay.
Don't jump the gun.
I wouldn't want to do that. That's right. So, welcome to the panel 5 fundamental physics session. That part works. My name is Carl Lineberger. I'm the moderator of this section. I expect that you will hear as little from me as possible as we go ahead, because we have an excellent group of people here who are much more capable than I. And one of the things I've learned from a long time ago, if that's the case, you sit back and be quiet. So we have 3 panelists today that are trying to span parts of what are covered in the full fundamental physics program. And the first panelist is Brian DeMarco, immediately on my right. Brian is, there we go. He does a variety of problems in condensed matter physics using ultra-cold atom lattice and other techniques, and a lot more. In his copious spare time, he's involved in a variety of DoD advisory programs. He's currently
he has a life somewhere else. The second speaker will be Eric Cornell, who will take more or less one topic and tell you more about it, where Brian's been trying to give you an overview. Eric, I just put BEC and much more. He was there at the beginning, enough said. And a major focus of the fundamental program now are these ultra cold and ultra cold. The third speaker is Ron Walsworth, and the best way to characterize Ron is certainly he does some precision measurements, clock things, but in looking at his background, it's hard to figure out what he doesn't do, and it's clear that it would have been much easier to tell you what he doesn't do than what he does do. And I don't know how you keep up with your group, Ron. But in any event, these three are going to be able to give you an excellent overview with a good case study of the fundamental physics program. I don't want to waste any more of their time, and so I'm going to turn it to Brian.
Thank you. Let's see if I can get this to work. I'll mention these actually. So, right. So there's a few set of recommendations, and I'll actually mention these in the overview I gave. Good. Okay. So I will, as Carl mentioned, I will try to give you a high-level overview of what's in the fundamental physics program and what makes it exciting, and what I think some of the challenges and opportunities are. So one of the recommendations in the decadal study consisted of identifying 4 areas of physics that were ripe for opportunities to make progress on microgravity. They're listed here, and what I'm going to do is really just tell you about 2 of these today. I'm going to talk just with the first one, which is the click, which is investigation of matter in the vicinity of what I call classical critical points. This is an example of an experiment that was done on the ISS. It's a program which has already yielded fruit, and it's allowed a range of parameters of classical liquid gas phase transitions to be explored that you can't achieve on Earth. So that's an example of something which has already yielded fruit. What I'll spend, and I'll come back to this at the end, what I'll spend most of my time today talking about, and all of us will, is CAL, which is a new program and is about to be launched in October, and it stands for the Cold Atom Lab. And it's an experiment on the ISS to create ultra-cold gases of atoms. So let me tell you a little bit about ultra-cold quantum gases. This is a picture from my lab, and I'm not going to show you any data or tell you about my research today, but I just want to explain that these are very complicated experiments. This is an 8 by 12 foot table crammed with hundreds of optics and about 15 lasers and magnetics, all focused down into a vacuum system which is hidden deep in the bowels of this thing. There's a glass cell there evacuated to about a picotorr, where we create these ultra-cold gases of atoms using laser cooling, laser trapping, and magnetic trapping. What are these substances? These substances are cooled to the lowest temperatures in the universe, just billionths of a degree above absolute zero, sort of 1 to 10 nanokelvin. They consist of about 10,000 to a million atoms. Typically, people work with alkali atoms. The size of these gases might be about 30 microns in diameter. They're very dilute. Maybe the highest densities are 10 to 13 or 10 to 14 per cubic centimeter. And these are the most quantum mechanical matter that we have on Earth. And so these experiments are a real playground to explore fundamental physics. So in fact, why are these exciting? The reason I love these kinds of experiments, and many other people do too, is because it allows us to reach really unsolved frontiers in physics. One of the biggest frontiers and most important, in my opinion, frontiers of 21st century physics is a quantum particle problem. It turns out that we don't know how to understand what happens. You put together many interacting quantum particles. These problems are not computable, and they span all domains and scales of physics, from neutron stars to the inside of protons and neutrons, of photosynthesis and electron transport, and high temperature superconductors. They're not computable. We can't put these problems on a computer. We'll never be able to do that. And we don't understand many aspects of them. So these ultra-cold quantum gases allow us to learn about the physics relevant to these problems and test our theories and approximations. They're also enabling tools that allow us to test fundamental properties, or as a physicist would say, in physics parlance, fundamental symmetries of nature. So, for example, ultra-cold atoms are our most precise clocks, and they allow us to try to study things like drift in the fine structure contest, fine structure, constant drift in time of how electromagnetic forces work, or even look to see if the speed of light is isotropic in space, because maybe that's not true. Actually, using atoms and interferometers, just like light interferometers, but using matter waves, is a way for us to hunt for new forms of dark energy and to do new kinds of equivalence principle tests. The equivalence principle is they inertial and gravitational mass is the same, and that may not be true, and we need to test that. So it is both a substance of fundamental interest and an enabling tool for us allowed to probe nature in new ways. So that's what's exciting about these kinds of experiments. What is the Cold Atom Lab? The Cold Atom Lab takes that giant experiment in my lab and crams it down into a volume that fits in a quad locker. That's an incredible achievement. It does it using something called atom chip technology, which we can talk more about if you're interested. This is the flight science module getting integrated, and some of the sort of middle circular feature there is about an inch in diameter to give you some sense of scale. And that's all getting fed into a quad locker now, which will be launched up onto the ISS in October. And so the idea is that this experiment, which will be, to my knowledge, the most complicated experiment done on the ISS, will be remotely operated through a K-band link, and it will be maintained by ISS astronauts, and they'll be trained on replacing parts of this as if they fail, and hopefully they don't. So what's exciting for me here is that microgravity will enable all kinds of physics that are new on this frontier. So for example, when you hear more about other kinds of physics, one thing I'm excited about is the opportunity in microgravity on the ISS to make a superfluid bubble. This is something you simply can't do on Earth because gravity always pulls all the fluid in the bubble down to the bottom and makes it into something which is not a bubble anymore. And it's really pushing what's possible in physics and pushing our knowledge of these kinds of strongly interacting quantum fluids. There are, so to give you a sense of the scale of the program, there are 5 flight investigations that are funded and getting ready to do science. They span areas such as few and many body physics, and Eric will talk a little bit more about that. These microgravity bubble superfluids, developing new atom interferometry tools, improving them in an environment like the ISS, which is not something we've done before. And also trying to figure out how to employ those to do new tests of general relativity or look for new forms of dark matter. There's a small ground PI program, so there's 2 funded ground investigations, one at Michigan to look at spectroscopy of Rydberg atoms, and another at Berkeley looking at what's called coherent magnon optics. And these are really their programs to develop the science and technology to form a basis for future investigations. Okay, so it's the 5-minute mark. That's good. I'm going to wrap up with another discussion of recommendations and then some comments. So there was, in the decadal report, there was 2 sets of recommendations that were sort of finite. One was to explore areas of physics, and that's happening now. There are either in all those 4 areas, or are there things undergoing, like DeClick, or there's something about to be launched, or plans in the works for future missions. There are also recommendations that are shown here. One was to have a ground-based program, and that exists. It's quite small. In my opinion, it's too small, and we really need to grow that so that there are future scientists and future science to be done. But you can check that off at some level. There was also a recommendation to do a rapid return to flight, and the reason I brought up DeClick is because from the point of view of Cal, it's a relatively simple experiment that's relatively inexpensive as well, and so that was one that could get up on the ISS quickly and yield fruit, and you saw data from that and physics that was explored. The third recommendation was that NASA should explore partnerships with other nations, and that's happening. DeClick is actually a collaboration with CNS. There's PK4, which is the Dusty Plasma Experiment, which is a collaboration with Roscosmos, and BCAL, which is a planned future follow-on mission that we hope to see at some point, maybe even the ISS, to follow up on Cal, sort of a more sophisticated version of Cal is being developed in collaboration with DLR. The fourth recommendation was to form a high-profile program that could draw in really extraordinary researchers, and Cal is an example of that. There are amongst those flight investigations, there are 3 Nobel laureates who are funded to do work on Cal. So that's another set of recommendations, and really progress has been made on all of those as well. So just to wrap up with a few thoughts, and let me gather my thoughts here because it's that point in the afternoon where it's hard to think. And I was saying I teach a class from 2 to 5 on Fridays, and let me tell you, that can be hard sometimes. So I think Cal is a great start. It's exciting because we can access new physics at the frontier of physics that we can't access on Earth. So I think that's fantastic. It's also an opportunity to learn about how to do these experiments in LEO and maybe even farther out in space. And it's nothing like this has really been attempted before.
The maximum leg break force is maintained with the exercise prescription I described to you before. So therefore, we see positive effect of that prescription on ISS in terms of this particular variable. When we look into aerobic capacity, you will see that it is also maintained on ISS, which is the green line and the orange line in a bed rest group doing a similar kind of prescription, but in the blue group, which is the control group with no exercise, you see a decrease in the aerobic capacity. So this prescription seems to work for some of our major variables, but we are now actually analyzing data for many other variables concerned with muscle and endurance training. This is a study done by our behavioral and science team in another element by Dr. Phils, looking into virtual motivation for exercise and behavior, looking into some virtual systems in order to have the astronauts motivated to doing exercise, because one concern we have for 2 to 3 year mission is that they might not like to do exercise 6 days a week for 2 years, the way it's being done in an isolated environment. So we need motivation. That's why I'm showing these slides, but it's more of the expertise of the rest of the panel. And also an intelligent personal trainer in order to motivate and instruct the astronauts to do the correct prescription that we have defined. The final 2022 deliverable will be what we call an integrated countermeasure suite, encountering all of the physiological countermeasures. I will not go through it systematically because I don't have the time, but this is a list of what we could consider each astronaut to be exposed to. However, we anticipate an individualized approach, so that some astronauts will do more of this and less of that, and it will vary from what type of person physiologically we are talking about, also behavior-wise. So we have pre-flight, in-flight monitoring, training and prevention, treatment and prevention, and then after planetary landing, certain physiological exercises. But I can go into more detail if there are any questions from the audience. An example will be that if you look into treatment and prevention, then anti-VIP, which is our highest prioritized risk, we might use, we don't know, Alan, but we might use bracelets and combine it maybe with dynamic exercise and trap blood, or using lower body negative pressure device or a combination in order to mitigate the fluid shift, but we still don't know exactly what we'll do because we haven't characterized the risk very. And then we have an alternative approach. I still have 1 minute, right? my time? Oh, okay. So, yes, artificial gravity will be considered, and this is actually because of the vision impairment problem on the fluid shift. So we are considering together with the engineers using one of the fuel tanks, an empty one, with inside rotations, that these are the green belts, and then having astronauts being exposed to a certain G level, which has to be defined. So we're trying through animal studies and human studies on the ground to define this threshold for G's. That has a huge impact on the habitat. So this has a high priority to determine whether we need this or not. Thank you.
Throw it open to the audience. Questions? Yes.
Thank you for this session. And I want to thank all the panelists. Actually, this has all been very interesting all day long. I have a question, which is more of a clarification question. It's based on information that I guess I had more intimate knowledge on many years ago. And that is, and it's regarding to sort of mostly to the teaming and personality issues and so forth. And the question in my mind came up because is my understanding, at least before, there was less than half of the individuals in the space station were from the U.S. I don't know what it is right now. And the and we had very limited or no information on the medical characteristics, the information in the background for the Russian cosmonauts. I don't know what information we had about the other team members on there, but I'm just looking at this because in the National Space Station, and the question really is, how do we really control the whole team in itself? Do we have veto power over people? And I guess that's probably not the case. But I'm just wondering, how does this work? And how do you really control the whole dynamics of the team when half the people or more you don't really have control over?
Steve?
Well, cultural diversity has been a major topic in the composition area because of that particular issue. And remember, I mentioned fault lines, where you have different people from different cultures, but also different disciplines within each I also talk about macro cognition and collaborative problem solving, which thrives on diversity because people from very different, diverse backgrounds can bring in different viewpoints. So rather than go to a selection strategy to create a more common team, I think we have to embrace or know that diversity because it brings in different perspectives which will create a thriving macro cognitive climate. However, the research by Sean Burke, funded by NASA, showed that cultural diversity actually led to more dissatisfaction, unlike what we see in the traditional work. I think there's a need for training and how to do collaborative teamwork, collective knowledge work with diverse groups, especially over long duration missions. So I'm not sure that composition approaches, selection of staffing approaches are going to be the best way to deal with that particular issue. I would look for more teamwork training that focuses specifically on that. And in the non-space literature, we've done quite a bit of that.
So can I just say, the NASA astronauts, they cross-train, of course, and they spend a considerable time in Russia. They're fluent in Russian, not just like conversational, but fluent, and vice versa. But that will address the team, the thing you said about teams in task performance, but not actually the team, the team dynamic building thing. That's right.
Excellent.
However you said that. Yeah.
Excellent point. Cross-training will facilitate the transactive memory and shared mental models, cognitive parts, but not the kinds of issues that would drive conflicts. So that was a good point.
Yes, Alan Hargens, UC San Diego. Related to these international crews, would it be worthwhile to do more analog environment studies to look at the selection and performance of the crews?
There's actually, I think, much of the research in the team area has used ground-based analogs, a number of them. So they've been doing that kind of work. They can't match, obviously, the long duration, but there have been, I think, 60 day mission, 60-day experiments for people in those environments.
And one year, the high seas
yeah.
is
so there is analog work going on.
And how do those data compare with what we have on space station?
I can't answer that specifically. Usually, I'm thinking back to the reports I've read. Often, those kinds of studies are paired with interview data from people who have been in the space station the conclusions are the combination of both. So I can't answer that particular question, but a lot of the findings that I mentioned, a number of them have been found in the analog setting. A lot of that work is still going on. The key point is, as I said, when you look back at the 2010, 2011 report, it reflected, frankly, an old view of genes, and an explosion of stuff has come out, and that's going on right now.
Peter.
But I think that's an excellent point.
Thank you. And then one question for Peter. I think there's evidence that we don't activate our stabilizing muscles sufficiently to give us like the activation of the normal daily activities for our skeletal and cardiovascular system. Is there any efforts that we need to do in order to address that?
Yes. The data that we are now analyzing from the SPRINT study, this prescription I described how to use the in-flight exercise equipment. It turns out that certain muscle groups are not maintained. We don't know what it means for balance post-flight, in particular when you land on a planetary surface or come back to Earth. And the reason that we will not give it a high priority is that the mission scenario right now is focused on a deep space Mars mission without landing. And when they come back to Earth, there's a host of people taking care of the astronauts. So we are not so concerned about that as the first step into deep space. But the next step when they land on Mars, there will be pivotal to understand that, so that they don't lose balance on the surface of Mars and will be able to do sensible things within a short period of time. But certain muscle groups are not maintained, as well as the back stabilizing muscles that we need to do more about. I would like to answer one question about psychology and behavior, which I'm not a specialist, but I actually know something, and that is that internationally Dr. Dinges has participated in the 530-day Mars mission, and he's also participating with the same measurements on the ISS, and will be able to compare data from those two missions, and an analog extended mission with the ISS, which is actually perfect, and I know he's doing that. And we are also, by in physiology, planning for 2 international collaborations in something called the NVHAP facility in Germany. 2 better studies are being planned and being executed within the next 2 years. So we are trying to do that international collaboration. It is we have been instructed by our director in the program to understand there will be a multicultural team probably going to Mars, at least when they land on Mars.
Hi, Rob Farrell from the committee in the University of Florida. I'll start with a question for Peter, but it's going to tie back with other things we've heard earlier in the day. That basically pull on the notion of genotypic suitability for, well, let's just say for exploration or space flight. But my first question is going to be, in the exercise physiological realm, we've heard things about other systems having to do with gender differences and other things that are sort of roughly genotypic. Are there gender differences in the sprint profiles that you would identify for people? And are you getting to the point where there might be individualized programs, sprint programs based on somebody's, eventually on somebody's genotype?
Yes. We are trying to implement that. The biggest obstacle to do genetic research in physiology is actually ourselves, because we have rules and ethics that we, therefore there's strict rules as to identification of the test astronauts. For obvious reasons, I'm not against it. I'm just saying that it is an obstacle, because in no time things will flow out to the internet, and astronauts will be recognized if results are not kept. So that's one concern. But we are doing that more and more, and actually right now we are trying to have a study approved looking into the relationship between the biomarkers, omics, and genetics, and the ability to perform certain strenuous tasks that are related to specific missions. And since we are doing certain of these tasks in order to have physiological predictors in terms of the muscle strength and aerobic capacity, we are trying to tie in omics, biomarker research to identify the individuals based on that. So we are doing that, but we haven't gotten there yet because the policy of NASA is very new in this area. In regard to gender differences, it's very interesting, a little bit provocative, but I think I'm allowed to be provocative even though I'm here on behalf of NASA. That is, there is a countermeasure, more or less 90% prohibition against WIP does exist. Fly females, they will not exhibit the vision impairment syndrome to the same degree as males. So if you fly females, that problem will be very little, maybe a non-existent problem. So we wouldn't need artificial gravity, we wouldn't need LBNP, and Alan Hagen's study wouldn't need it to be completed, which of course I'm joking, but certain females do exhibit some signs, but not to the same degree as males. So that's a big gender difference. We have been looking into genetics in that particular syndrome in astronauts in space, and we see some aggregation of alleles that are associated with manifestation of vision changes and physiological changes, which has been published by Scott Smith and his group. It's very interesting. It's still being debated to the accuracy of what he found and the interpretation. So, yes, we are doing exactly what you are suggesting, and there are gender differences in certain of the highest risk, highest profile risk we have.
And before I yield the microphone for Andy and Stephen, is there a version of that discussion that happens in the psychological realm? I realize it would be much less correlative because of simply the 80-20 you were talking about, Andy. But what's the current thinking outside of NASA even on those correlations?
Well, several of us think that you're going to need to do some kind of genomic analysis in space. So you're going to have to, like, for people who were there, you're going to want to get, say, some sequencing done, epigenomic profile, at least gene expression measurements, because there are going to be convergent points. Like, people have a lot of genetic variants, but it's very difficult to sort out how they contribute to common phenotypes. But gene expression is a different matter, and so you might be able to get early indicative markers for problems that might develop, probably more so for physiological measurements, things like, muscle physiology. You could think about doing things like muscle biopsies and then sequencing for gene expression or for DNA methylation. Things like that seem pretty reasonable, and the technology for doing sequencing in space is almost here. The sort of miniaturized sequencing, like the ion, thing is, that sort of thing is doable. There was there was sequencing done by Kate when she was in space, not of astronauts, right? This is some prepared but she's shown that just preparing materials to do genomics and so forth is just not difficult at all in the space environment. The one thing that's a little tricky, the last step of preparing DNA, you need to do some kind of a ethanol or isopropanol precipitation, and that's just absolutely out. But there are ways you can use absorptive compounds to purify the DNA at the last step. That's one little thing that would probably have to be worked out.
Great.
Okay, thank you. Last, one more question? Wait, we have somebody up at the mic? Yes? Okay.
You can go. Mike Piccolo, Loma University, ASGSR board member. Let's pretend you have the perfect crew that you chose on the ground. You figured out all the dynamics and everything. As soon as you go into space, you're going to have immune changes, you're going to have CNS impacts due to radiation. Are you thinking about those problems? It's going to change those dynamics just because immune impacts behavior and vice versa.
Steve.
And I've sat in several of the sessions there, which we had the radiation people and the sleep people and so forth talking about it. And we've been raising questions to say, you're all talking about sleep and radiation impairments on the individual, but recognizing the individual as part of a group, and these are the and sleep cycles, and these are the specific ways that they're going to start to pay to function. So when I was interested when you said we have the perfect crew. For me, the perfect crew is going to be resilient and adaptive and be able to handle those kinds of mini shocks, micro shocks to the team system. However, I think in the last year when I was attending some of those meetings, we were raising questions such as you thinking about cross areas where the sleep research you have and the sleep findings you have and the radiation findings you have, put them in a team context. If you're talking about what they do to the individual, and don't think about the individual just as one member of the team, think about how those impairments on the individual in turn influence macro-cognitive and collaborative problem-solving processes. So that is, we've been pushing that question for them to address more and more, and that calls for a collaboration across a multi-team system, if you will, in that particular environment.
Yes. This will be a last question in order for us to stay on schedule.
I can ask it separately. There's not.
No, please. Yes.
So beautiful panel discussion. Thank you for Peter and for any of the other panel members. With regard to integrating the gender differences potentially into your data,
are you considering looking at the differences in the microbiome? Because at least in animals, not in humans yet, to my knowledge, there's evidence of a microbiome that's specific to males and females, and males and females obviously have different susceptibilities to different diseases or physiological attributes. We were lucky; we actually had two females out of the 6 in our blood studies. We thought that was terrific. But I'm just wondering if this could be of help in predictive medicine and personalized medicine as you're going forward with trying to integrate this information into your studies.
The answer would be yes from my side. This is the first time I hear about gender specifically to the microbiome. We haven't done much microbiome. We have done one study on by Dr. Lorenzi on the ISS microbiome study looking into those aspects, and these data being analyzed and presented for the time being. We are doing another study, planning another study in an isolation study at our facility at Johnson Space Center called HERA, and that will also be a microbiome study. But the researchers, to my knowledge, are not focusing on gender differences in this particular context. It depends. On the ISS, we have very few females being flown, and I have suggested to the chief scientist some time ago, why don't we fly more females? But this is a political problem, and in the isolation study we have, it depends on what kind of subjects we can recruit, because we have some problems doing that. So right now it's probably difficult, but something probably should be done.
Especially since hormones regulate the microbiome and all kinds of things, right? So it could be of relevance.
We should be open to that.
Okay, well, thank you, panel. Thank you very much.
You want to start?
Yeah, I'll start.
Okay, everyone. We've reached the part of the agenda where we're going to reach out and one more round of try to get some additional community input while we have everyone here. But first, before we do that, first of all, Rob and I would like to say thank you to all the panelists It was absolutely fantastic. I know I learned a tremendous amount today, and greatly appreciate you taking the time to prepare and come and do this along with the questions. I think we had an absolutely fantastic day. So thank you again to all the speakers. And these things don't get pulled off just because a few of us show up today. I would personally like to thank Sandra, Deanna, and Marcel for all the work they did behind the scenes to help pull all this off. So thank you. So as far as input, what we'd like to do is we're going to open up the floor here in a second to get whatever input you guys have for us as part of the committee's activities on this review. Rob and I are going to be taking notes. This is all recorded, so what you give us, Sandra's taking notes. We'll be able to have the notes recorded and available to the committee. The committee members have all been taking notes. I've seen that all day. So please feel free to give us your input. Also, for those of you that may still be online, is you can use the email address microgravitymidterm 2016 at nas.edu to give us input. The sooner you give it to us, the more useful it will be. Don't wait till August. And also recognize that if you do give us input through the email address, it's part of the public record also, and we'll deal with it in that way. So to kind of kick off this discussion, this morning Gail Allen gave us the overview of where the agency is, or where NASA is in terms of exploration planning, and the gateway notion, the gateway approach is relatively new. I think it was first unveiled to the NASA Advisory Council about 3 or 4 weeks ago. So recognizing that that's new, I guess the first question I've got is, how many of you heard it for the first time today? That's what we figured, is that there'd be a fair amount of that. So it was really good that we did that. One of the things that we noticed, or that I've picked up on, is that the gateway like it's far away. In the late 2020s, when we start putting HABs and other things up in orbit, but that's only 10 years away. And as in my former life, a hardware development kind of guy, 10 years really scares me, knowing what the R&D schedule turns out to be, independent of any budget conversation. So given that context, I guess some of the input we would like to get is, what do you folks see as some of the tent poles, the long tent poles, maybe the shorter tent poles, to help make that gateway approach happen? And frankly, any other thoughts? And Rob, feel free to add or subtract from what I just said.
Okay. No, seriously, folks, I think the questions that have been brought up today, yeah, we've heard and we're keeping track of and everything, but there are certainly things that we learned today that we didn't know until we walked in. For example, that the gateway actually has form and function. We know that it's size now that we didn't know before. These are things that impact the discussions that we're having. So, sort of the questions that, well, first off, we want to collect everything that we can. If you walk away from here, even if you're in the room, let alone if you're online, you can email us at that, and we'll get it into the record. But for the purposes of discussion today, what did you learn, or what did you hear that changed